It's going to be a long session. So, Assalamu alaikum, everyone. My name is Ashaz Sayeed. I'm a second year medical student, and I'll be teaching you guys basically all of histology for this course. Okay. If you have any questions, you can mail me at asaeed@alfaisal.edu, and any questions you can send me on WhatsApp at my number here 053-952-368. And so I will be covering in two sections. First, I'll cover the cardiology section, and then I'll cover the pulmonology section. And the cardiology section first, I will cover some basics you have to know for understanding the next three lectures. Then in the pulmonology lecture session, I'll go through some basics in pulmonology, and then the next two lectures. So we'll start. Okay, so our cardiovascular system works in a way of pumps and pipes. Okay, so it's like a water pump, you may say. The pump being our heart, and the pipes being the blood vessels. Okay, and obviously the fluid being the blood. So first we'll talk about the heart. The heart itself, if you guys are not familiar till now, it has four chambers: the two atrium and then the two ventricle. Um, it has four valves: the tricuspid valve and the mitral valve, and it has the pulmonary and the aortic valve, which transports blood from the ventricles into the superior blood vessels. Okay. And the cardiovascular system, you can divide it in three layers. So over like broadly, the cardiovascular system is in three layers. Okay. So we have our tunica externa, the tunica media, and the tunica intima. Okay. The tunica externa is the outermost layer. Okay. If you think it as a pipe, the outermost layer of this pipe is going to be the tunica externa. The middle layer is going to be the tunica media, and the innermost layer is going to be the tunica intima. Okay. So inside the tunica intima, you're just going to have the lumen. Now, if we talk about the external part, the tunica externa is the outermost layer. It's made up of connective tissue, and its function is to connect the blood vessel to the rest of the body and keep it in place. The tunica media is the middle layer. Okay, it has it has muscles in it, and this musculature is for contracting the heart or the blood vessel. Okay, so this contraction is the reason why blood moves in our body, in like the cardiovascular system. So remember this MMM. It's for tunica media as the middle layer, and it's the muscle layer. So it's just three M's. Now you have tunica intima. I for intima and I for innermost. So it's the innermost layer, and it consists of a single layer of simple squamous epithelium for most of the time. Okay. So under the epithelium, like this is the epithelium. Inside of it is going to be the lumen right here. So under the epithelium, you're going to have connective tissue. Like just under the epithelium, you have the basement membrane on which the epithelial cells lie. And then you're going to have a connective tissue. So you can see right here, this red line where my arrow is pointing at, if you guys can see it, that is the epithelium with the basement membrane, and the pink region under it, that's where your connective tissue lies. And the thing about tunica intima is that this is the only layer which is present in capillaries. Your bigger blood vessels, they're gonna have all three layers. But in capillaries, you're gonna have only the tunica. Okay. Now let's talk about the tree principle. So from the heart, you're gonna start off with a huge artery. It's called the aorta. It's the biggest artery. Okay. Now this aorta, it starts off thick and less in number. Then it branches off. So the aorta, it becomes thinner while it branches. Okay. So arteries, they are thinner and they are more in number. And then these arteries branch out into something called arterioles. Arterioles are even greater in number, but in thickness they are much smaller. And then we reach capillaries. Capillaries are the greatest in number, but size-wise they're pretty small. Okay. Now from here, so we go from the aorta all the way to the capillaries. From here, you're gonna have venules fusing into each other to form the vena cava. We have uh, the superior vena cava and the inferior vena cava. These two are the biggest veins in the body. Okay, so just see the diagram. It starts off as a single aorta. It branches into multiple capillaries and then it merges back into vena cava. So all I want you guys to know from here is arteries start off thick and they branch out into the tissues. Veins, they start at tissues. Many people have this misconception that veins start at heart. No, the veins start at the tissues where the arteries ended. From there, they're gonna merge in towards the heart. Okay. And functions of the CVS, uh, it pumps blood, obviously. Okay. And remember this, it pumps blood in one direction only, in the forward direction, which means arteries are gonna carry blood from the heart towards the system, towards the other tissues and all that. And from there, veins are gonna carry blood from the system back to the heart. It's never gonna be possible that veins are gonna carry blood from the heart to the system. You get what I mean? And other functions include oxygen and nutrient exchange. You're going to have temperature maintenance and transparent delivery of hormones and immunity cells. Okay. Cool. Now, this slide, I just added it for your information. It just has a bunch of prefixes and suffix and all that. So you don't have to memorize everything on it. It's just for a good read. Okay. So let's start with heart histology. Do you guys have any questions so far? Okay. So, so we'll start off with the histology of the heart. So we'll talk about the blood vessels and the capillaries later. For now, it's just the heart. Okay. We'll start off with some basic definitions. Uh, the heart has cardiac muscle cells. Okay. So these are like special cells. They are not smooth muscle cells. They are not skeletal cells. They are a cell of their own. Cardiac muscles. They are present only in the heart. They're also known as a cardiomyocyte. And whenever you hear cardiomyocyte, it refers to a single cardiac muscle cell. Okay. Now, a cardiac muscle fiber is basically when you have many of these cardiomyocytes back to back on each other. Okay. So cardiomyocyte 1, cardiomyocyte 2, cardiomyocyte 3, they line up on each other and they join each other by something called intercalated discs. More about this later. Okay. Cardiac muscle tissue is just a general term to describe a bunch of cardiac muscle cells. And you guys have to remember this. morphological syncytium and a physiological syncytium. Now, first I'll tell you guys what syncytium is. Syncytium is just like many smaller units, they come together to work as one big unit. That's what syncytium is. Basically, cells working in unity. Okay, many cells doing one function. That's a syncytium. It's of two types. Morphological. So here the cells actually merge into each other. They're gonna share the nuclei, and it's gonna appear like one cell. So many smaller cells merge into each other, and they form this one giant cell with many nuclei. Okay, something like this is seen in the skeletal muscles. Physiological syncytium, here the cells don't merge into each other. They are just in contact. They're just in contact. They're at contact at the points of intercalated discs. So it's like a cell and another cell. They touch each other, and here you're gonna have an intercalated disc. Okay, and through this, they're gonna send signals to each other to contract in unison. So it's written here, cells don't merge into each other. They're connected by intercalated discs for them to work together. And something like this is seen uh, a lot in the cardiac muscle. Okay? Layers of the heart. In the basics, I was telling you guys about how everything in the cardiovascular system is divided into three layers. Okay, the tunica externa, the tunica media, and the tunica interna. Right. Well, you don't really have it named as tunic, or you don't have a tunica externa in the heart. They have different names in the heart. Okay. So we have three layers as I discussed before. We have the pericardium, which is analogous to the tunica externa. We have the myocardium, which is analogous to tunica media. And we have the endocardium, which is analogous to the tunica intima. Okay. Um, Here you can see an image of all the muscle layers in the heart. The yellow thing you can see over here, the yellow thing, that's the epicardium. It's like it's above the pericardium. Okay, and it's just adipose tissue. It just fixes it around on the surrounding surfaces. Okay, and then you have the pericardium. Under this yellow tissue layer, you have something called fibrous pericardium. Okay, and under that you have parietal pericardium. Then you have the pericardial cavity. Then you have the visceral pericardium. Then the biggest chunk of layering. So you have this layer like a sandwich. The thickest portion, that's the myocardium. Okay, the myocardium. That's the thickest portion. And then finally you have the endocardium, inside of which you have finally reached inside the heart. Okay. So outside the pericardium, it's just attachment to the surroundings. And inside the endocardium, you have the lumen of the heart, where you will actually see blood within the heart. Okay. And we'll start with the pericardium. Pericardium is of two types, serous pericardium and fibrous pericardium. Okay. So if this is going more towards the heart, more inside the heart, the fibrous pericardium comes first. Okay. And then the serous pericardium. So check this. First, we start on the fibrous pericardium, and then we move into the parietal pericardium. Mind you guys that the parietal and the visceral pericardium, both of them are part of the serous pericardium. Okay, remember this. Parietal and visceral, both of them are a part of the serous pericardium. Okay, so here we got first we start at fibrous, and then we move into the parietal pericardium, which is also known as the outer serous pericardium. Then you have the pericardial space. Okay, this space it's filled with something called pericardial fluid, and this fluid it basically prevents friction and provides support to the heart. Because if this fluid is not present, the heart will get damaged due to friction. Okay, because the heart beats all the time, it should not scrub against a rough surface when it beats. Because at every beat, when it scrubs against a rough surface, you can have scar tissue formation, and that will cause damage to the heart. So just to keep an insulation to prevent it from uh, getting damaged by friction, you have the pericardial fluid. Okay. Then you have something called as the visceral pericardium. The other name for this is the epicardium. Okay. Remember this name. Visceral pericardium is equal to the epicardium. They are the same thing. Don't get confused. Okay. So these are some diseases associated with the pericardium. It's not really that important, I would say. Know it. The most important, I would say, is cardiac tamponade. The others are not really relevant. So if you'd see in the image over here, you have the fibrous pericardium, okay, and then you have the parietal layer of the CS pericardium. Inside of it, you have the pericardial cavity, which is filled with pericardial fluid. Inside of that, you're gonna have the epicardium, which is also known as the visceral pericardium. And once all of this is done, uh, like this is all of your pericardium, okay? All of this, right in the image you can see here, all of that is the pericardium. Under this, you're gonna have the myocardium, okay? So we'll talk about the pericardium a bit more. So you will be given some histology slides, okay? And you have to identify whether it's the epicardium or not. So to identify that you are at the epicardial level, you see some blood vessels and nerves sitting in between fat, okay? So it's gonna be like this bed of fat inside of which you're gonna see blood vessels and nerves, okay? See the image here. This entire thickness right here, that that is the adipose tissue. And then this circular structure you can see, this circular structure, that's a coronary artery. So that's a blood vessel. Okay. You can see a nerve here. The difference between a blood vessel and a nerve is that blood vessels will be empty from the center. Nerves will be completely full. You can see here. The blood vessels are empty and the nerves, they're full. Okay. If we talk about the structure of the epicardium, it's gonna be having first of all, it will have the simple squamous mesothelium. Okay. Its shape is simple. Okay, so it's not layered, it's one layer, it's squamous, and it's called the mesothelium. Okay. Under this, you're gonna have the basement membrane. Under this, you're gonna have areolar connective tissue and the adipose tissue. Okay. So you guys can see here. First, you guys have the mesothelium, which is written M in the image. The image it's written M, that's the mesothelium. Under it, you can't really see the basement membrane here because it's really thin. Okay. Under that, you're gonna have your connective tissue and fat. This is the connective tissue, and then under that, you're gonna have this fat right here, this white colored fat. Okay. So that's all about epicardium. We are done with the pericardium. We are done with the epicardium. Let's move into myocardium. Okay. Myocardium, it lies under. Okay. It lies under the epicardium, and we are getting more close to the lumen. So how to identify? You can see striated muscle cells, okay, arranged in parallel. You can see here. You can see the image. The cells themselves, they are striated, which means they have like dark and pale alternations on them, okay, and they have something called intercalated discs. So you have something called intercalated disc right here, okay, here too. So these intercalated discs are a characteristic of the myocardium. Okay, if you want to differentiate between skeletal muscle and cardiac muscle because they look pretty similar, you have to find out about these intercalated discs. Okay, If they are present, then it's myocardium or cardiac muscle. And if it's absent, it's a skeletal muscle. Okay. So we're done with that. The structure. Here, what happens is the cardiomyocyte. You can see this 1 second. You can see the two red lines, and then you can see a cardiomyocyte inside of it, right? So just above this line, you have something called the intercalated disc right here. This connects another, what do you call a cardiomyocyte, okay? So they work together in a physiological syncytium, as I told before. Capillaries are present, but they're not really visible in the slides. I think there's one. There, this thing right here. Okay. That's a capillary. Okay, and remember dyad feature. It's not really important, but remember. Okay, in skeletal muscle cells, you have a triad. Here you'll have a dyad. The details about this, you guys probably took that in physiology. Okay, just remember that there's this difference between the cardiac muscle and the skeletal muscle. Okay, the location obviously is the middle layer. It's sandwiched between the pericardium and the endocardium, and these are some diseases associated with it. You have myocarditis, you have blood vessel disease causing cardiomyocyte injury or death. Basically means if there is blockage of blood vessels which supply the heart, that will cause injury and death to the cardiomyocytes themselves because they're not getting adequate nutrition. Okay, and then you have conduction system. Next. So we'll first. I have this thing called spot the difference. Okay, I created this so that you guys can differentiate between skeletal muscle and cardiac muscle easily. So it has alternating points which you can use to differentiate them. Okay. First, we'll talk about uh, the skeletal muscle. Skeletal muscle, it's multinucleated and it has a morphological sensation. Okay, as we talked before. And the nuclei here, they are pushed towards the plasma membrane of the cell. Okay. Good. So the nuclei, they are pushed towards the sarcolemma. See the image here. The nuclei, they are not at the center. Okay. They are at the sarcolemma. They are pushed at the ends. Look at this. Okay. The nuclei are pushed to the ends. You don't see intercalated discs here. You don't see them at all. And they branch less often. Okay. Cardiac muscle, on the other hand, they are a uni or a binucleated physiological sensation. So again, as I told before, cells join on each other in the presence of intercalated discs. Okay. We have another one here. And the nucleus, it's more on the center. We can see this thing, right? If it was on the periphery, like the skeletal muscle, the nucleus should have been here, but then it's not here. It's not there. It's at the center. Okay. Intercalated discs are present. So anything, if you'd forget everything, the one thing I would like you guys to remember is if you want to differentiate between skeletal muscle and cardiac muscle, just look for the ICDs. Okay. Just look for them. Just look for the intercalated discs. If you find them, it's the cardiac muscle. If you don't, it's the skeletal muscle. Okay. Same thing here. It's just that was a longitudinal section on the side, and it's a transverse section from the top. Okay. So here you're gonna see pink dots. Okay. The skeletal muscle, you're gonna see these pink dots right here. And for cardiac muscle, you won't see dots. You're gonna see like this smudge-like appearance. Okay, it's like you ran over them. Okay, you see like a smudge appearance. Okay, and the skeletal muscle, you see a proper dot-like appearances. So, so far I've been telling you guys so much about the intercalated discs, and you might be wondering, what is it? What is it made of? Okay, remember four structures it's made up of. Okay, four structures. First is the interdigitating sarcolemmas. Now to explain you that, it's just, let's say this is cardiomyocyte 1, and this is cardiomyocyte 2. Okay, and sarcolemma basically means a plasma membrane. So the plasma membrane of the first cardiomyocyte, it goes very close to the plasma membrane of the second cardiomyocyte. Okay, in the shape of the staircase, you can see in the diagram here. See here, see this. This, that's your sarcolemma of one cell. This thing is the sarcolemma of the other cell. Okay, so you can see how it's in like a staircase pattern, right? This looks like one step of a staircase. Okay, now we have adherence junctions. Adherence junctions are basically these 2 points which help these cells hold together. Okay, so we have cell 1 and cell 2. For them to hold together and to not fall apart, okay, you have two structures, the fascia adherens and the macula adherens. Remember them. Okay, the fascia adherens and the macula adherens, they work to keep both of these cardiomyocytes together. Okay, and now finally we have gap junctions. These are responsible for the physiological sensation we see. Okay, for example, let me mark them for you. For all the cells to contract at the same time, they should have a signal going through them at the same time. Okay, so this signal is transmitted from one cell to another very quickly through these gap junctions. Okay, if these gap junctions are not there, the cells, they don't contract together, they contract independently. That's going to be a problem. We want these cells to contract together, produce a force big enough to push blood from the heart into the blood vessels. Okay, so for them to contract together, the signals you need move through these gap junctions. So that's why the gap junctions are responsible for the physiological sensation. Okay, so we are done with the pericardium, we are done with the epicardium, we are done with the myocardium, and finally now we are the endocardium. This is the innermost layer. Okay, inside of this you're gonna see the loop. So this is the innermost layer, and the biggest identification for the endocardium. If you want to search for the endocardium, you search for Purkinje muscle cells. Okay, these right here, and these things right here. Okay? These are your Purkinje fibers. These are like white or pink. It really depends. Okay, here they're pink. In some places they're gonna be a bit white. Okay? So to identify the endocardium, you search for the Purkinje fibers. Okay. To identify cardiac muscle cells, you search for the intercalated discs. To identify endocardium, you search the Purkinje fibers. Okay. For the structure, you have a simple squamous endothelium that had what do you call it? The epicardium, it had a mesothelium. The endocardium has an endothelium. Okay. Now, wherever there is an endothelium, there is gonna be a basement membrane. Okay. And areolar connective tissue. This, all of this. That's gonna be your areolar connective tissue. There are gonna be some fibroblasts within there and Purkinje fibers. Okay. Remember the structure. it's gonna be uh, your endothelium, under it, basement membrane, under it, you're gonna have the connective tissue and the Purkinje fibers. Four things you have to do. Location is the innermost, obviously, and it faces the cardiac view. And function, the endothelium is important. First of all, it's for oxygen exchange because uh, oxygen from the blood goes from here to supply the Purkinje fibers and to supply the cardiomyocytes, which are close to the lumen. The cardiomyocytes, which are farther away from the lumen, they are supplied by coronary arteries and veins, okay? But which are closer, the nutrients can just go through by diffusion. So that's one thing, oxygen exchange, okay? And the other important thing is the endothelium produces proteins and factors which prevent clotting of blood, okay? It's important. It's because if you have a blood clot forming in the heart, okay, and if this blood clot moves away from the heart into the lungs, then the person can die immediately. Okay, yeah. So clot formation is really bad, and the endothelium prevents. Just remember. Okay. Again, spotting the difference between the epicardium and the endocardium. Okay. In the epicardium, if you have a slide of an epicardium, you're gonna see in the slide of an epicardium, you're going to see these nerves right here. This is a nerve, okay? And this is a blood vessel. This is a blood vessel, okay? It's within the adipose tissue, as we told before, okay? So anytime you want to identify the epicardium, you look for these structures, okay? If you want to differentiate it from the endocardium. The endocardium, you have your Purkinje fibers, okay? These things. So anytime you want to differentiate between epicardium and endocardium, the one thing I want you guys to remember is for the endocardium, you search for Purkinje cells. For the epicardium, you search for blood vessels and nerves lodged within the adipose tissue. Okay. Let's go. Purkinje fibers. We have been talking a lot about the Purkinje fibers. Okay. And this is just a detail on them and what they do. Okay. So to identify them, you just see these large cells, these large cellular structures in the endocardium. Okay. Remember that these are non-contractile. They do not contract. Okay. And they are specialized cardiomyocytes. So the regular cardiomyocyte, it contracts. This is a special cardiomyocyte. It does not contract, and it's a pacemaker, which means uh, signals from this signals from the Purkinje fibers go into the cardiomyocyte and they determine the rate of contraction. Okay. To us, know that it's non-contractile. Okay. And the location is just under the endocardium. Okay. It's like more specifically just under the endothelium. It's part of the endocardium, but it's just under the endothelium. This might sound a bit confusing, but I'm sorry. I don't put it right. Okay. It's just under the endothelium and it's in the endocardium. Okay. Function. As I just said, it can create its own action potential on which the cardiomyocytes will contract. That's one thing. But its main function is conducting action potential, which started at the senoarterial node, then it goes into the AV node, then through the bundle of His, and then into the fibers. In case you have your SA and AV nodes failing, that's when the Purkinje fibers start their autorhythmic function. It's not always, okay? Normally, normally, they are just conducting action potential, okay? So now, again, another spot the difference between cardiomyocyte and a Purkinje fiber. So cardiomyocytes are usually smaller in diameter. They're more compact. Okay, they're much more compact. They're smaller in diameter. On the other hand, Purkinje fibers they look more large. Look at this. This is much more compact. You can see that the empty cytoplasm it's not really much as compared to a Purkinje fiber. You can see it's a huge cell. Okay, it's a huge cell and it's more light in color. Both of them have desmosomes, but cardiomyocyte again, cardiomyocyte always has the ICDs, the intercalated discs, and this is what you're gonna use to differentiate them. Just engrave this in your mind. Okay, cardiomyocyte, ICD. Purkinje fibers, it's usually not present. It's debatable. Okay, some say it's present, some say it's not. It's not an important feature. ICD is important cardiomyocytes, not Purkinje fibers. Okay, cardiomyocytes they are more pinkish in color. Purkinje fibers they are more pale or white. Okay, and cardiomyocytes you're gonna have T tubules, and Purkinje fibers you don't have T tubules. Okay, just a moment. Okay, okay. So all this time I've been telling you guys that the Purkinje fibers they conduct action potential. Okay, now this action potential has to reach a muscle cell, a cardiac muscle cell, in order for the muscle cell to contract. Okay, goes from here, the action potential goes to the muscle cell, and the muscle cell contracts. Okay, so the point at which the action potential moves from the Purkinje cells to the muscle cells, it's called the Purkinje myocyte junction. Okay, there's a point where the Purkinje fibers connect with the cardiomyocyte and they send signals to them, which come from the autorhythmic nodes. The autorhythmic nodes being your SA node, seating arterial node, and your AV nodes. Okay, signals which come from there conducted into the Purkinje fibers, and now the Purkinje fibers via the Purkinje myocyte junction transfer this action potential into the cardiomyocytes for them to contract. Got it? So we are done with the endocardium, we are done with the myocardium, we are done with the epicardium, pericardium, we are done with the Purkinje fibers. I think this is the last structure you have to know, valves. Okay. So we have, as I told you before, we have four heart valves. We have the mitral valve, the tricuspid valve, the aortic valve, and the pulmonary valve. Okay. Remember this one thing. Okay. Valves are avascular. Many people, I've seen that. Many people make this mistake of thinking that valves have blood vessels in them. No, valves are avascular. They have no blood supply. Okay. And you may ask, if they don't have blood supply, how are the cells within the valves alive? See, valves are present in the lumen of the heart, okay? And the nutrients in the blood, they just diffuse inside, okay? Just that. So we have two structures in valves, two important structures. Remember them, because this thing, the fibroelastic leaf, this was asked in the previous year questions, okay? This was asked in the previous batches. It's really important, remember. Now, the fibroelastic leaf, it's like the main flap structure. So the valve has like three flaps this way, right? So the main leaf-like structure of the valve, that's the fibroelastic leaf, okay? You can see in this diagram, this thing right here, this is the fibroelastic leaf. It's surrounded by endocardium cells, okay? Surrounded by endocardium. So the cells of the endocardium and all that stuff is there. And then at the base, just here, just at the base, you have the myocytes. You don't really have to remember the structure all that much. They probably won't ask that. Just remember that you have two structures involved, the fibroelastic leaf and the chordae tendini. Okay. It's a description. It's really not that. Just Remember, please remember these two structures, fibroelastic leaf and chordae tendon. Okay, in this image right here, this this is the fibroelastic leaf. Okay, this right here, that's the fibroelastic leaf. The chordae tendini, just by their names, chordae, cord, tendini, they look like tendons. Okay, so these structures right here, these structures, these are your chordae tendini. Okay, and they connect themselves to the papillary muscles. And that's all you have to know. Just know about fibroelastic leaf, about chordae tendini, and you should be good. Avascular, fibroelastic leaf, chordae tendini. These three things involves. Remember them. Okay. The fibrous skeleton. Okay. Yeah, this is the image. This image right here. These are the four valves. Okay. You can see this. These are the valves. These are the valves. Okay. You can see this thing right here, which is in between the valves. You know, this thing in between the valves. That's the fibrous skeleton. Okay. The fibrous skeleton, it surrounds the valves. Okay. And it's made up of collagen. Remember the fibrous skeleton, it's made up of collagen and it's not conductive. So any action potential, it does not it does not affect the fibrous skeleton. Okay. So uh, but somehow the action potential has to cross the fibrous skeleton to supply it to the ventricular walls. Okay. Action potential can cross only via something called the bundle of Higgs. Okay. Remember these 2 points over here from this slide. Remember these 2 points. Action potential can cross via bundle of His. That's one thing. And this delay is responsible for the atrial ventricular contraction because the atrium, they contract first and then the ventricles contract. Okay. Atria contract to push the blood in the ventricles. Ventricles contract to push the blood into the blood vessels. So this delay is because of the presence of this fibrous skeleton. It stops the action potential for some time. Okay. So it has to be crossing only via the bundle of His. Remember this point. Bundle of His, fibrous skeleton, action potential. Just remember that. Okay. Then you have, oh yeah, this thing. Okay. So this is the conduction pathway, how the electrical signals move in the heart. Okay. Again, the name intrinsic nervous system. Our actual nervous system, which we have like from the brain and the spinal cord and all that, it's not this one. Okay. Our brain is not consciously controlling this because it's its own nervous system. Okay. It's a separate, its own nervous system. And if we talk about how it goes, the sinoarterial node, this has pacemaker cells. Okay. So they generate an action potential. Now, how do they generate this action potential? You guys took that in physiology. Okay. So this right here, the sinoarterial node, action potential is generated here. From here, they now move towards this structure, which is known as the AV node. Okay. So we move from the sinoarterial node, action potential is generated in the sinoarterial node. From there, the action potential moves to the AV node. Okay. Now from here, we are moving into this thing. Okay. This thing right here. That's where the bundle of His starts, which we talked about in the previous slide. Okay. The fibrous skeleton is located somewhere around here. Okay. It's located somewhere around here. And signals have to cross this fibrous layer. They have to cross it. And the pathway for them is this bundle of His right here, this thing. Okay. From here, it branches off into the left and right bundle branches. Now, these are your bundle branches. Okay, and finally, it moves from the bundle branches towards the apex, and from the apex, it moves upwards. Okay, so it came here, from here, it goes down all the way here, and now it moves in this direction. Okay, sinoatrial node, AV node, bundle of his, bundle branches, and then up the Purkinje fibers to supply the ventricular. Okay, done with that. Second. These are the nodes right here. Okay. Moving from the sinoatrial node, and then the AV node, and then the bundle of his, and then the bundle branches, and finally the Purkinje fibers. Okay. So now we have something identify. Just identify the structures. Okay. Give me an answer for what this structure is and what this structure is. Write it in the chat, or you can unmute and say it. No worries. This one and then this one. Guys, come on, write in the chat. What's this image and what's this image? Or you can unmute it too. Okay, so no one seems to know it. Okay, yeah. Bottom is cardiomyocytes, yes, but uh, the first one, B is actually not the endocardium. Okay, yeah, bottom is cardiomyocytes. That's correct. That's correct. Okay, this one is cardiomyocytes, but this one it's not endocardium. Actually, it's the mesothelium. Again, whenever I'm telling you guys, whenever you want to search for the endocardium, see Purkinje fibers. Okay, here I don't see any Purkinje fibers. I don't see any Purkinje fibers here. Okay, it's just this white hue with some mesothelial cells over there. Okay, so it's the mesothelium. Notice the absence of Purkinje fibers. So engrave this, put this in your mind. If you are searching for the endocardium, look for the Purkinje fibers. If they are there, then that's the endocardium. If it's not there, then that's the epicardium. Okay. Or sorry, not the epicardium. Okay. Here's the epicardium. This one you guys were right because the integrated discs that with the main cable. Okay. And we are done with the first lecture. You guys have any questions so far? How to distinguish between Purkinje fibers and adipocytes? They look a bit similar. See, first of all, if you have like a histology slide, okay, Purkinje fibers, they are more close to each other, okay, and they are more pinkish in color. Adipocytes, they are white. They are absolutely white. Adipocytes are 100% white. It's like nothing's there over here. Okay. Purkinje fibers have this slight pink color. Okay. So no more questions for this lecture. Do you guys have any questions? Oh, okay. So let's move on to the next one. So lecture two.
Recording in progress.
Lecture two. That's gonna be blood vessel histology. So far, we discussed about the heart, Now we'll be talking about the histology of the blood vessels. Hopefully, it's gonna be a bit easier. Okay, the heart, I get it, it's like a bit complicated. This one's it's much more easy. Okay, and start. We're done with the heart. Now we'll be talking about the blood vessels. Okay, you have the arteries, you have the veins, and connecting them, you have the capillaries. So it's all just basic. It's all just basic. The arteries that conduct oxygenated blood. Remember this, it's really basic. Arteries that conduct oxygenated blood from the heart to the system, to like the tissues and all that. And veins take deoxygenated blood from the tissues back to the heart. Okay. Arteries, oxygenated blood, veins, deoxygenated. Okay. Now the arteries, they branch out, as I discussed before in the basics. Arteries start less in number. They branch out to form millions of capillaries, and from there, veins actually start in millions, but then they merge together to form the biggest veins, which is the inferior vena cava and the superior vena cava. Okay, they're opposite. So, with that, just remember these words: arteries branch out, veins merge. Arteries branch out, veins merge. Remember that. Okay. Yeah, here you have some more differences. Arteries, they again branch out, veins merge. Okay. Arteries, they have three tunica layers and they have a thick tunica media. Okay. Veins, on the other hand, they have three layers again, but the tunica media is thin and they have warms. Okay. You have your thick tunica media, you have a thin tunica media here, plus you have warms. Remember this characteristic of veins, the presence of warms. Arteries, they do not have warms. Veins, they have warms. Remember this, okay? Arteries, they carry oxygenated blood. Veins, they carry deoxygenated blood, okay? And this is the pathway. Arteries, they start at the left ventricle at the aorta. They start at the aorta, and now they branch into capillaries, okay? Veins, they start from capillaries, and they merge into the superior vena cava and the inferior vena cava, and then the right atrium. Arteries, they start off less and their number increases. Veins, they start off more and their number decreases. Okay. And we talked about arteries, we talked about veins. Now we'll talk about something called lymphatic vessels. Lymphatic vessels are pretty similar to veins in structure. Their function is different. Okay. Their function is to carry like some interstitial fluid back into the circulation. Okay. But structure-wise, they're pretty similar to veins. Okay. They too are of three layers. They also have a thin tunica media and they also have walls. Okay. They follow the merging pattern just like veins. They have three tunic layers similar to veins and they have walls. Okay. The lymphatic vessels, they merge just like the veins. They merge into two main ducts, the thoracic duct and the right lymphatic duct. Okay. The thoracic duct and the right lymphatic duct. These are like the final big ones in the lymphatic system. Okay. And these two finally drain into the subclavian vein. So we have your thoracic duct and your right lymphatic duct, and they drain into the subclavian vein. They don't drain in the heart. Remember this. Some people get this confused too. Okay. They don't drain in the heart, they drain in the subclavian vein. Okay. So whenever there are like major blood vessels, okay, you're gonna have your arteries, your veins, your lymph vessels, and nerves. They're all gonna be together in a big bundle, okay? And these are just some rules you have to remember if you want to name them. It's not really significant for the MCQ part, okay? Just know that it's gonna be easier for you guys to identify, okay? So all these structures, your artery, your vein, your lymphatic vessel, and nerves, these are all parallel to each other in a bundle, okay? And for the arteries and the veins, the blood flow is in opposite directions. So it's like a two-way highway. You know, when you have a highway, the road, and some of the cars move in this direction, some of the cars on that side move in the opposite direction. It's the same way. You have opposite blood flow in arteries and veins. Okay. And now, uh, the arteries and veins. So we have the arteries, they become capillaries, and then from the capillaries, they move to veins. So both of them are continuous at the capillary beds, and all of them have similar names. If we'll see this example right here, okay, in the femoral triangle, you don't have to know what the femoral triangle is, okay, but just see that the nerve, the artery, and the vein, all of them have the same name, femoral, okay. So now we'll classify blood vessels. We talked about arteries, we talked about veins, we talked about capillaries. Now within arteries, let's classify them based on the tunica media, okay. So what the tunica media contains, the classification is on that, okay. So the tunica media in blood vessels, they obviously they have the smooth muscles. Okay. Also, they have something called fenestrated elastic laminae or sheets. Now, these elastic sheets are the reason why your blood vessels can dilate or constrict. Okay, they're elastic because of these, and the number decides the types. Basically, the number of layers for the smooth muscle cells and the number of layers of elastic sheets. That's what decides which type is it. Okay. So let's start with arteries. So type 1 artery. Type 1 artery. Now you have the type 1 artery. Type 1 artery, usually they are the biggest ones. Okay. They are the biggest ones. They're pretty easily visible. Okay. The structural name is an elastic artery because they're pretty elastic, and functional name is the conducting artery. Okay, remember these names because they might ask questions in these alternating names. Okay, they won't mention type 1 artery or they won't mention just elastic artery. They might ask you something related to a conducting artery, and you guys should know what a conducting artery is. Okay, now for identification, you have more elastin lamellae. Okay, you have more of these elastic layers than you have smooth muscle layers. Okay, you can see the image over here. See this thing? Okay, the pink hue you can see over here. That's your smooth muscle layer. Okay, now this black structure right here. You can see this black structure right here, right? That's gonna be your elastic layer. Okay, your elastic laminae. So you have your pink smooth muscle laminae, and then you have your black elastic laminae. Okay. The tunica intima, it's the same. It's the same thing I'm telling you guys. You have the endothelium, under you have the basement membrane, the connective tissue. It should be engraved in your heads, okay? You have the endothelium, and this endothelium, it supports itself on the basement membrane, and under the basement membrane, you have connective tissue. It's everywhere, okay? Tunica media, know these important points, okay? In the tunica media, the elastic layers, you have 40 to 70 elastic layers, okay? And you have only 30 to 40 smooth muscle layers, so almost half, okay? You have 40 to 70 elastic layers and only 30 to 40 smooth muscle layers. And you do not have fibroblasts, okay? This thing is gonna be, the fibroblast thing is gonna be an important determining characteristic for, green arteries, okay? Now the tunica externa, the outermost layer, it has collagen. And now this thing, nerve and blood vessels. Okay, it's like NVB stands for neurovascular bundle, NVB, and it's with vasa vasorum and nervi vasorum. Now, what is vasa vasorum and nervi vasorum? Okay, see your arteries. Okay, especially the aorta. It's a pretty thick artery. Its wall thickness is also huge. Okay, and you cannot what do you call distribute nutrients via diffusion in such a thick wall. Okay, you need extra blood vessels within the blood vessels to supply the wall. You get me? See, for example, this is like a huge blood vessel. Okay, it has a thick wall. Okay, and the blood within the blood vessel, it's not enough. The diffusion from nutrients, the diffusion of the nutrients from this blood, it's not enough. Okay, so for that, you need this neurovascular bundle. It's like a small neurovascular bundle within the walls of a blood vessel. Okay, vasa vasorum literally means vessel of a vessel, and nervi vasorum is the nerve of a vessel. Okay, and the elastic lamina in tunica intima, it's not for type 1 artery. It will come later. Okay, but for we are talking about the type 1 artery, and here, uh, most of the elastic layers they're in the tunica media. Okay, yeah, I know the type 2 and type 3 have some of it in the intima, but here mainly it's gonna be in the tunica media. Okay, now function it's expands and now it's like a huge blood vessel. It's close to the heart, and a good example of this is aorta. Aorta is the best example for type 1 artery. Okay, the biggest differentiating example, the best example, because it's the thickest artery and its analogous vein, you know, its counterpart. It's the superior vena cava and the inferior vena cava. Everything else is going to be almost pretty similar. Just remember the points I've highlighted because those are mainly the differentiating points. Okay. Type 2 artery. Type 2 artery, it's known as a muscular artery because here now you have more of smooth muscle. Here you have less elasticity and more smooth muscle. Okay. Its functional name is distributing artery, structural name is muscular artery. Know that, just know that. Okay. And what do you call? Yeah. For identification, you have two, only two fenestrated elastic laminae. Okay. In type 1 artery, you had 40 to 70. So from 40 to 70, we drop down to just two. Now, since they are that less, we can give them names. Okay. So we have the internal elastic lamina. See the diagram. You have the internal elastic lamina, and then you have the external elastic lamina. Okay. Just two. So if you see something like this, see the diagram, you see like these two dark lines, you see these dark lines, and between them it's this light grayish color. Okay, you're probably looking at you are looking at a type 2 artery. Okay, so here, if you was asking, does the tunica intima have elastic layers? It was not there in type 1, but now it's there in type 2. Because in the type 2, the tunica intima, you're gonna have the internal elastic lamina. Okay, you can see this convoluted thing right here, this thing. Okay, whatever that is. So that's the internal elastic lamina, and it's in the tunica intima. Okay, uh, now it's like it's in between the tunica intima and media. Okay, but we classify it in tunica intima and tunica media. Well, elastic. See, we have only two elastic layers in type 2 artery. Type 2 artery, two elastic layers. Okay. One of them is like at the starting of the tunica media and where the intima ends. Okay. And the other layer, it's in the external elastic lamina. Okay. And this thing is in the tunica media. Okay. The internal layer, it's at the boundary of the intima and media, and the external layer, it's in the tunica media. Okay. Tunica externa, it's the same thing as type 1 artery. It also has this neurovascular bundle with vasa vasorum and nervi vasorum for the same function to supply nutrients to the wall of the artery. Okay, the function here is distribution of blood. Okay, so now the blood has moved from the aorta. Now it's gonna go into the major parts like your arms and legs. Okay, and it's a bit further away from the heart. A good example here is the ulnar artery. Okay, now remember this example. Just remember, they will probably ask this for OSPI. Okay, it's analogous vein. Obviously, it has the same name, so ulnar vein. Okay, ulnar artery example. This example, ulnar artery. It's important for OSPI. Okay, they can put this image right here. Then they can ask you which artery this might be. Is the ulnar artery because it's a type 2 artery. Okay, now we have the type 3 artery. We are getting smaller and smaller. Type 1 was the biggest. Type 2 is a bit small. It's more in number than the type. Type 3 is even smaller and it's more in number than type 2. Okay. It's the same thing. You just add small at the end. One second. Small, small. Small muscular artery, small distributing artery. Okay. The identification here is one elastic lamina. We started at 70. We dropped it down to 2. And now in type 3 arteries, we are at 1. Okay. See this layer? This convoluted layer right here. That's your internal elastic lamina. That's the only one is there. You don't have an external elastic lamina. Okay. So structure, obviously you have the tunica intima, tunica media, and the tunica externa. Okay. The same thing. Uh, the tunica intima has the internal elastic lamina. Tunica media, it has 10 smooth muscle layers and no fibroblasts. Okay. In comparison, your type 2 artery, it had 20 smooth muscle layers and it also had fibroblasts. Remember this, I forgot to mention it, I think. But remember this. 20 smooth muscle layers, 2 elastic layers, and presence of fibroblasts. And out of the 4 types of arteries we have, only the type 2 artery has fibroblasts. It's a feature of just the type 2 arteries, the presence of fibroblasts. Okay. Type 3. Uh, this has 10 smooth muscle layers, one elastic lamina, which is the internal elastic lamina, and it does not have fibroblasts again. Okay. The tunica externa is the same thing. You have the neurovascular bundle with vasovasorum to supply all of this because all of this, it's still a pretty thick layer and it still needs, even after becoming so small, it still needs a neurovascular bundle with vasovasorum and nervi vasorum. Okay. Again, the function is distribution of blood is further away from the heart. And examples, digital arteries, the arteries within your fingers. Okay. So just again, last time, remember 10 smooth muscle layers, no fibroblasts, and one elastic lamina. Okay. Finally, we reach the type 4 artery. It's not even called artery at this point. It's called arteriole. Okay. Its functional name. Now, this is an important functional name. Okay. All the other functional names, they were just man. But this functional name, the resistance artery, is an important. Okay. Again, this thing has no elastic lamina. Look at this. Look at this. There is no elastic lamina. Okay. This thing is just one smooth muscle cell. It's just rounding the whole artery. Okay. You can see here, it's like just 1 to 2 smooth muscle cells, not much. Okay. Tunica intima, again, is the same thing. You have endothelium, basement membrane, connective tissue, same thing everywhere. The type 4 tunica intima, it does not have the internal elastic laminae. Type 4 itself does not have any elastic lamina at all. Okay. So it just has the endothelium, this thing. You can see this thing. Okay. So there's the endothelium and it bulges. Okay. During vasoconstriction, what happens is the flat endothelium, it becomes engorged. Okay. To decrease the vessel diameter. That causes vasoconstriction. Okay. Again, no elastic layers, 1 to 2 smooth muscle layers, and it has no fibroblasts at all. Okay. This thing, now here, the vessel is too small. Okay. Type 1, type 2, type 3. These three types, they had their own neurovascular bundle with the vasa vasorum and nervi vasorum and all that. Type 4 artery, it's really small, really, really small. Okay? It does not need its own neurovascular bundle. You can see here, see the image. It's just like 6 cells. That's enough. That's enough for diffusion. Okay. Its function is important. Its function is really important. Okay. Blood pressure control. Till now, type 1, type 2, type 3, all of them, they just had what do you call transporting blood. Okay. Type 4 is important for blood pressure control. Okay. So the arterial blood pressure is controlled by the contraction of type 4 arteries. Okay. And obviously, from here now you're gonna enter capillaries. Type 1, type 2, type 3, type 4. And from type 4, you're gonna be entering capillaries. Okay. Example, there are too many to name. They're in the millions. They're in millions. Okay. Fibroblast and tunica externa. No, no. Fibroblasts are just in the tunica media. Okay. Tunica externa does not have fibroblasts. Uh, yeah, I think we're done. Yeah. This is an important thing. Two signs of vasoconstriction. Okay. So we have the endothelium bulging in the lumen, and then you have corkscrew nucleus of the small vessel. Just remember these two. Okay. We are done with arteries. Now we have capillaries and then the veins. In capillaries, we just have the tunica intima and the tunica media. The tunica media, yeah, the tunica media is replaced by something called pericytes. I'll talk about it later. Okay. Veins are also classified similarly. They also have valves. Okay. And valves are avascular. Nothing much. Veins. Uh, the thing you have to remember about veins is that structurally they're pretty similar to arteries. It's just that when you reach the bigger veins, such as the superior vena cava and the inferior vena cava, here we have a new feature. The tunica intima is just like the arteries. The tunica media, composition wise, it's like the arteries, okay, but it's thinner in average. Tunica externa, it's thicker, okay. Also, the tunica externa has these longitudinal smooth muscle layers. Till now, the smooth muscle layers we had were, you know, they were surrounding the thing. Now, in the tunica externa, we have these straight running smooth muscle layers. Okay. Now, these contract and they prevent collapse of the SVC because arteries, they have high pressure. Okay. Veins, they don't have much pressure. So, yeah, also remember this, the tunica externa, that is the layer which has the longitudinal smooth muscle layers. Okay. The tunica media also has smooth muscle, okay, but the special longitudinal smooth muscle layers which prevent collapse of the blood vessel, that's then tunica externa, okay. Many people will confuse this for having for it being in the tunica media, but no, it's in the tunica externa. Just because you have smooth muscle layer doesn't mean they're always in tunica media. Just don't get confused, okay. And I have been telling you guys all this time that arteries have thick tunica media and veins have a thin tunica media. Ever wonder why? It's simple because arteries, they carry blood with much more pressure, and to support that pressure they need a thicker muscle. Veins, they don't have that much pressure, so for that they don't need a thick muscle wall. Okay, they need like a small muscle wall. That's done. This is a really important image right here. Okay, this was asked previously. Okay, so this describes a neurovascular bundle, and this is a vein. This is a small vein. Okay, how can I know it's a vein? Look at the smooth muscle layer. It's pretty thin. Okay, the external layer, it's much thicker. Okay, and the shape, it looks collapsed. Arteries, on the other hand, the muscle layer, It's much thick. Okay, look at this. It's much thicker. Okay, it has elastic lamina. In this case, it just has the internal elastic lamina. So it means that this is a small artery. Okay, it's a type 3 artery. It's not a type 2. Type 2, you're gonna have an external elastic lamina. But no, it's just one lamina. So this is a type 3 artery. Okay, and whenever you want to differentiate between blood vessel and a nerve, you see blood vessel has this lumen in there. Okay, when this has a lumen, nerve, it does not have a lumen. It's a solid structure. Okay. Okay. Now identify these. Identify the first image and the second image. Let's see who gets it right this time. You can unmute and say it too. You can type in the chat. Okay. What's the first structure and the second structure? Type. What's this? What's this? This is a type 1 artery. How? You can see alternate dark and light bands in there. We see mixed dark and light lines. The dark lines being the elastic laminae and uh, the more pinkish lines being the smooth muscle. Okay, now this one is the superior vena cava. How? Once again, this is a huge, what do you call external layer. Okay, the tunica external. And the tunica media is pretty small. Okay, so that's why it's the SVC. And we are done with this lecture. You guys have any questions for this lecture? Any questions? Now we are in the last lecture for what do you call cardiovascular system. Okay, after this we'll start the pulmonary system. Okay, we have the microvasculature histology, histology for the capillaries. Okay, okay. Microcirculation is the place of U-turn back to the heart. So till now, what was the load? We went from the arteries, we go to the arteries, take a U-turn, become veins, and go to the heart. Okay. So the arteries they change into veins, and every cell needs a capillary supply for nutrients. And capillaries again for like the 10th time. It just has the tunica intima surrounded by pericytes. And since they don't have a tunica media, we classify capillaries based on how permeable they are. But before we talk about capillaries, let's talk about the endothelium. Okay. Endothelium within the capillaries is just this simple squamous cells and they're aligned in the direction of blood flow. Okay. And function is diffusion, exchange of gases, okay, anticoagulant and anti-bleed. These two are important and overlooked functions. They won't ask this targeted the exam, but then it's just for you guys to know. Endothelium it produces factors which prevents platelet formation, plate formation. It prevents coagulation. Okay, because if you have coagulation, the blood clot can block blood flow. Okay, and that's going to be a problem. So it prevents coagulation and it also prevents bleeding because if you bleed, you bleed. Okay, so it prevents all that. Now it's everywhere in the CVS, it's everywhere in the cardiovascular system. And this is a microscopy. This image is important, I feel. Okay, because of the presence of these bodies, these are known as the Weber Palade bodies. Just know their name, how their function. It's not really significant for what do you call it? For histology, at least. So these bodies right here, those are Weber palette bodies, these dark bodies. Okay. And these things are something called venocytotic vesicles, and huge molecules are transferred through these vesicles. Okay. Just have this picture, printed in it. The Weber palette bodies and the venocytotic vesicles. So we're done with endothelium. Now let's finally get into capillaries. Okay. Structure, only tunica intima for like the 12th time with endothelium, basement membrane. Remember, it does not have subendothelial connective tissue because capillaries are pretty thin. They're pretty thin. Okay. So they can't, they actually can't afford to have any thickness to them because here diffusion of nutrients happen. Okay. Diffusion of oxygen happen. So we minimize the thickness. Okay. Specialties, it's thinnest, so it's vulnerable to high blood pressure. So if someone has high blood pressure, capillaries can explode and the person can have bleeding. Okay, they're not the largest in diameter, but combined capillaries are the longest. If you take every single capillary in our body and then you put them back to back, that's gonna be longer than any aorta or any blood vessel we have. Okay. Um, this thing, this thing is important. Okay, if you want to identify capillaries, okay, you see, look at the cursor. Okay, look at this. Look at this thing. Okay, over here too. You can see these cells in there. These cells. Okay, these are red blood cells in the capillaries, and you can see how they are back to back. Okay, they are not mixed. It's like one RBC here, one RBC here, one RBC here. It tells us two things. One is that the capillary, the thickness of the capillary is just as big as the red blood cell. That's it, red blood cell. Okay. One red blood cell is pretty small, and the thickness of capillaries is as thick as that, if not even thinner. Okay. So capillaries are pretty small. And the other thing is that they line on back to back. First of all, they don't have space to be standing side to side. So they are back and front. Okay. The second thing, this minimizes distance for the diffusion of oxygen. Okay. So if you see a slide in the longitudinal view and you see that you have RBCs back to back standing in a queue. Okay. That's going to be capillary. Over here, you won't see you won't see line. You'll just see one red blood vessel. Okay. That's because this view, it's like you cut the capillary and then you see it from the top. Okay. That's why it looks like there's only one blood vessel. But behind this, you have yeah, one RBC. Behind this, you have many more RBCs. Okay. Again, remember, RBCs stand in a queue. Next one, capillary bed. Okay. Now, this thing is when you have a lot of capillaries in one place, and a true capillary bed is always RGB. It's a mnemonic for you guys because uh, people who use laptops and play video games a lot, they know RGB. It's on color. Okay, so R is red, which represents arteries. Green is G is green, which represents lymph, and B is blue, which represents vein. Second, look at the diagram here. Look at the diagram. This side, okay, that's the artery side. Okay, that's red. This is blue. Okay, and this right here is green for the lymphatics. Okay, now we have some new structures you guys have to know in context of capillaries. Okay, we have something called a pre-capillary smooth muscle sphincter. Big name. Let me tell you what it does. Okay, so you know how it's an arteriole and then you have the capillaries. Okay, it's not necessary that blood will go through every single capillary in the capillary bed. Okay, the capillary bed is a collection of many capillaries, and it's not necessary that blood should go through every single one of those capillaries in the bed. Okay, so what determines how much blood goes and how many capillary beds are open? Okay, it's these sphincters. So think that this is a smooth muscle sphincter, a pre-capillary smooth muscle sphincter, and think that my arm, this is the arteriole, and these fingers, these are the capillaries, okay? So if this smooth muscle contracts, it blocks blood flow, and these things, they will not be active, okay? So blood does not flow. And if these smooth muscle sphincters relax, blood flows, okay? Now we have something called meta-arterioles. Meta-arterioles, they are between the arteriole and the capillary, okay? Like the transition zone. So look at the diagram here. This was an arteriole. This was an arteriole, okay? These are capillaries, okay? This small region right here, just a branch of, that's the meta-artery, okay? It's basically a connection between the arterioles and the capillaries, okay? And it comes after the arterioles, as I told you. It comes after arterioles and before the capillary bed, okay? It regulates blood flow to capillary beds by pre-capillary smooth muscle sphincters, okay? So look at this, okay? This is a smooth muscle sphincter. This is a smooth muscle sphincter, okay? This right here, that's the meta artery, okay? So if the sphincters are open, if they're relaxed, blood goes from the arteriole through the meta arteriole into the capillary bed, okay? Now, what happens if the uh, what do you call, what happens if the smooth muscle sphincters are contracted? You cannot let blood, just stay there. The blood has to move. Again, meta-arteriole comes into play. Because what happens here is, look at this. The meta-arteriole starts here, okay? It actually continues and joins the venule, okay? It's in this region, it becomes something called a thoroughfare channel, but you guys don't have to know that, okay? Just for the sake of simplicity, know that it's just a meta-arteriole. And in a closed capillary bed, it's a direct connection between the arterioles and venules. This is an important. Okay. This is an important. This is an important point right here. Is that in a closed capillary bed, the blood moves directly from the arterioles into the venules via the meta arterioles. Okay, it's an important point. So from this slide, the one thing I would like you guys to remember is that these two structures, the presence of the precapillary smooth muscle sphincters, the sphincters, okay, and the other thing, the meta arterioles, okay, and the fact that they are a direct connection between the arteriole and venule in a closed capillary bed. If some question asks you, how does blood flow from the arteriole to the venule if the precapillary smooth muscle sphincters are closed? The answer is via meta-arterioles. Okay. Also, the the smooth muscle sphincters, they are only at the arterial side. They are not at the venous side. Okay, they are only here at the arterial side. Okay. So that's all it for this slide. So again, just to add on there, open capillary bed, blood moves from the arteriole into the meta-arteriole, into the capillary bed, and then into the post-capillary venue. Okay, the sphincters here are relaxed, and the capillary bed is open in regions where you need high blood demand. For example, in exercise, your muscles need a lot of blood. Okay, so these capillaries open, supplying a lot of blood to the smooth muscles and not the skeletal muscles. Okay, so capillary opens, open capillary bed supplies a lot of blood to the skeletal muscles, and that's when you see like a pump. Whenever you go to the gym, people who go to the gym usually they say that I have a pump or something. It's because of these capillaries opening and supplying a lot of blood there. Okay, and the exact opposite of this is in the closed capillary bed, where the blood moves from the arteriole through the meta-arteriole and straight into the post-capillary venule. It does not involve the capillary bed. Here, the sphincters are usually contracted, and places where you don't need a lot of blood, that's where you have a closed capillary bed. Closed. Very small mistake. Closed capillary. Okay. Now we have lymphatic capillaries. Okay. Lymphatic capillaries, structure-wise, they are similar to veins. Okay, they start at the lymphatic capillaries. Okay, they drain into the thoracic duct and the right lymphatic duct, which in turn drains into the subclavian vein. Okay, now since they don't form a complete circle, okay, see how it's like arteries. Second, okay. So you have arteries becoming capillaries, becoming veins, and the blood going back into the arteries. So a cycle is complete over here. Okay, that's why it's called closed circulation. Okay, the regular blood moving in, that's closed circulation. Lymphatics, on the other hand, is an open circulation. Okay, why? They start at the capillaries. Okay, they drain into the thoracic duct. Okay, and then the right lymphatic duct, and this vein right here, that's going to be a subclavian vein. Okay, but from here you do not have some new lymphatic blood vessels which come back to the capillaries. Okay, that's not there. So they just drain. They start at the capillaries, drain. Start capillaries, drain. A cycle is not complete over here. That's why it's called an open system. Okay. Now there are some places you have to know which do not have lymphatic drainage. So, for example, the central nervous system that does not have lymphatic drainage. The tough structures in your body, like your bone, teeth, and cartilage, they don't have lymphatic drainage. Okay. The placenta does not have lymphatic drainage. The cornea does not have lymphatic drainage, and your epidermis. Epidermis is just the uppermost layer. It's already a dead layer, and it's avascular. It's just dead cells. They don't need lymphatics anyway. Okay. So just remember this. Okay. Just remember this. Clinical metastasis and edema. Okay. So cancer spreads through the lymphatic vessel. Okay. So that's how metastasis is basically the spread of cancer, and it spreads through the lymph nodes. Okay. Edema, it's when your lymphatic vessel fails to do its work, and now uh, you have water or fluid in the interstitium. Okay. So people who have edema, they usually have swollen hands. Okay. A difference between blood capillary and lymph capillary. Okay, blood capillary, blood capillary is thin and one-layered endothelium with basement membrane. You have a basement membrane in blood capillary. Lymphatic capillaries without basement membrane. Forgot to mention this, but blood capillaries, they have basement membrane. Lymphatic capillary, they don't have basement membrane. Okay, yeah, the bigger lymphatic vessels, the bigger vessels, they might have a basement membrane, but when we talk about capillaries, there is no basement membrane for the lymphatic capillaries. Okay, blood capillary, you have a smaller lumen. Lymphatic capillary, you have a large lumen. Okay, look at this. Look at this. Okay, it's a pretty small lumen. The gap in the lumen, that's pretty small. Lymphatic capillary, you can see how big this is. Okay, it's irregularly shaped and it's much bigger. Look at the size difference. Okay, look at the size difference. That's one thing. These are more regular shape. You can see it's more of like oval. Lymphatic capillaries, God knows what shape this is. Okay, and you have uh, tight junctions in the blood capillary. You do not have tight junctions in lymphatic capillaries. Okay, lymphatic capillaries are more permeable as compared to blood capillaries. Why? Is because they don't have the tight junctions. Just know this concept. Okay, they're more permeable because they don't have tight junctions. Blood vessels or the blood capillaries, they have tight junctions. Yes, they are permeable, but not as permeable as lymph or the lymph capillaries. Okay. Now we're done talking about them. We'll classify capillaries. Okay. classification is based on how leaky they are. Okay, since they don't have a tunica media, you have to classify them based on how much they leak. You know how permeable they are. Okay, we have three types. We have the continuous, we have fenestrated, and we have sinusoids. More about this in the coming slides. Okay, just know that continuous. Okay, that's the most common type and is the least permeable. Then comes fenestrated, a bit more permeable, and then comes sinusoid. Sinusoid is the most permeable. Okay. Uh, Now, before that, let's talk about something called the parasites. I've been telling you guys how capillaries have the tunica intima, and instead of the tunica media, they have parasites. Now we'll talk about what parasites are. Okay, so parasites. There we go. See the cursor. Okay, these cells right here. Okay, with the flat nucleus. That's your endothelial cells. Okay, the flat cells. Anywhere you see some flat cell is endothelial cell. Okay. Now this cell where the arrow is pointed to, okay, that's a parasite. Okay, and parasite is basically supporting the endothelium. Okay, its other name is a Roget cell. Okay, and it's a mesenchymal adult stem cell. So its other name is a Roget cell, and it also acts like a stem cell. Okay. Now, as for structure, they are cells with processes that try to surround the circumference of the capillary, and these cells and their functions are controlled basically by something called nitric oxide. NO is a short form of nitric oxide. Know that, okay? Now, about the processes, it's written processes. Look at this. Okay, look here. Uh, these things, these things, the cylindrical cells right here. Okay, these are your parasites. Okay, and these things, you see how it's going everywhere, trying to surround the cell. Okay, it originated here and it's trying to surround the circumference of the capillary. Now these things, those are your processes. Okay, these things are your processes right here. Okay, function. The functions of parasites is important, especially the ones which I'll be telling you guys to remember. Okay, first up, know that these are your contractile cells. Now, obviously, in the other blood vessels, you had your tunica media, okay, and the tunica media, it had smooth muscle cells which helped in contraction. Now we don't have that here, so we have something called we have the parasites which act as your contractile cells. Okay, they play a role in blood-brain barrier regulation. Okay, so just know that. Okay, wherever there's blood-brain barrier, you have a lot of parasites. Okay, this is an important function. Wound healing. Okay, so if someone gets a wound, okay, uh, since capillaries are pretty small, in a wound, capillaries can be devastated. They can be absolutely destroyed. And parasites are responsible for regenerating the structures in the capillaries. So the parasites give rise to endothelial cells. Okay, and in some cases they give rise to smooth muscle cells too. But mostly, they give rise to endothelial cells. Okay. Mhm. I think that's it. Location, parasites are actually located in the continuous capillary and the post-capillary venules. So this is where you're going to see the smooth muscle cells. The post-capillary venules, they have some smooth muscle cells. Okay. So parasites give rise to endothelium and smooth muscle cells in the post-capillary venule. Post, after. Capillary. Venule, a small vein. So after the capillary ends and it goes into a venule, that's your post-capillary venule. And that has smooth muscle cells. Okay. And as for capillaries, it gives rise only to the endothelial cells because they don't have smooth muscle cells. So it gives rise only to the endothelial cells. Okay. And its location is also, it's sandwiched between the endothelium and the basement membrane. This point, it's easy to remember because it's really funny how you have the endothelium here, and endothelium usually rests on the basement membrane so far, but then the parasite just enters and invades it. So now, instead of it being your endothelium and the basement membrane. You have your endothelium, parasite, then the basement membrane. Okay. Now we'll start with the three types of capillaries. First, we'll talk about the continuous capillaries. Okay. Now these ones, they have other names too. You have the somatic capillary. It's the least leaky. Okay. Does not leak. Okay. The somatic capillary. It's the least leaky of them. Okay. Now, you have for identification for the continuous capillaries. Okay. For yeah, there you go. So the continuous capillary, this is the continuous capillary. The other name, you have something called a somatic capillary, and these are the least leaky. And for identification, you see a full basement membrane. The shape is well defined, and you have really close parasites. Okay, look at this. Look at the shape of this thing. It's pretty well defined. Okay, it's pretty well defined, like a proper circle. It has parasites in there. Okay, and the basement membrane is complete. As for the structure, the endothelium, uh, you have the endothelium, you have the parasite, and then you have the basement membrane in order. So it's first the endothelium, okay, then you have the parasite, then you have the basement membrane. The endothelial cells, the cells themselves, okay, uh, they have like a complete basement membrane, okay, they have a complete solid cytoplasm. Okay? Now I'll talk to you guys later about what do I mean by solid cytoplasm or what do I mean by complete cytoplasm, because the other ones which are not as which are more permeable, let's say, their cytoplasm is actually porous. Okay, but for continuous capillaries, you have complete basement membrane and cytoplasm. Also, remember about the marginal folds with tight junctions. Okay, continuous capillaries are again they're highly regulated, they're less leaky, so they're gonna have these marginal folds right here. You can see this thing, which have tight junctions in them to prevent leakage of what do you call leakage of proteins and stuff. Okay, and as for function, small molecules can move through, just small, really small, less than 10 kilodalton. Okay, simple diffusion of small molecules, and if you want slightly larger molecules, you have to transfer them via vesicles. Okay, and they are the most common one. Remember this. Remember this. Okay, engrave this information in your mind. The most important or the most common capillary is the continuous capillary. Okay, and they're important in what do you call the blood-brain barrier. See, okay, so in the blood-brain barrier, they're really important. We're done with that. Let's go. So now in fenestrated capillaries, you're gonna see a full basement membrane. Yes, the basement membrane is gonna be full, but then the cytoplasm, it's not gonna be complete. The continuous one, it had a complete cytoplasm. In fenestrated, you're not gonna have complete cytoplasm because the cytoplasm will have these holes, you could say, called fenestra. Okay, and these holes are covered with a really, really thin membrane called the diaphragm. Okay, so we see a full basement membrane with parasites are rare. Okay, whenever you see parasites, they are commonly in continuous capillaries. Okay, these ones, it's rare. You will most likely not see parasites, fenestrated capillaries. Okay, most likely you won't see parasites, and you'll see some indentations in the wall. Now, what do I mean by indentations in the wall? Okay, look at this. Look at this. Look at where the arrows are pointing at. Okay, look at the electron microscopy image. You can see that there are some dents around the perimeter. Okay, so these dents are actually your fenestra, or like these holes which have been dug in the cytoplasm, and these are covered with a diaphragm. Okay, so yeah, endothelium plus basement membrane. Parasites are not really present. Let's assume that they're not there. Okay, so you just have endothelium and the basement membrane, and the endothelium, it has a complete basement membrane. Yes, a complete basement membrane. Okay, but the cytoplasm, you know, is fenestrated. It has pores in it, and these fenestra are covered with a diaphragm which is made up of glycocalyx. Okay, and the function is just faster transport of macromolecules. And location is in endocrine glands and tissues which have a lot of absorptions like kidney and intestines. Okay, so all I want you guys to remember from this slide is that uh, fenestrated capillaries, they have a full basement membrane, but they have pores in the cytoplasm which are covered with a diaphragm made up of glycocalyx. Okay, and it's located, like these capillaries are located in endocrine glands and in places which have a lot of absorption. For example, in the intestines and the kidney. Okay, and here let's final one, the final, the most leaky is the sinusoidal capillary. It's also known as sinusoid. You can commonly call it sinusoid. Okay, and here there is no chance at all. You're gonna be seeing parasites. Okay, uh, structure is again you just have endothelium with basement membrane, and here the basement membrane is discontinuous and the cytoplasm is discontinuous. Not only does it have fenestra, fenestra is small pores. Here the cytoplasm is just discontinuous. It's like huge holes in the cytoplasm, huge holes in the basement membrane, huge holes in the cytoplasm. Okay, and the fenestra of the cytoplasm, these holes, they don't have a diaphragm on them. Your fenestrated capillaries, they had smaller holes with diaphragms on them. These ones, they are bigger holes and they don't have diaphragms. Okay, they're very leaky, and these capillaries can actually, they can also cause the leakiness of cells. Even cells can move outside them. The first two capillaries, there was no shot that cells are moving out. Yes, some proteins, but cells no. Sinusoidal capillaries, you actually have cells moving outside. Okay, and as for the location, these are located mainly in four places. Okay, four important places which need its characteristics. Okay, so remember this, remember this, remember this, remember this. I think it was in some previous practice question or previous year question or something like that. Of where is the sinusoidal cavity located? Okay, just remember places which need a lot of permeability. That's where you're gonna find sinusoidal cavity. Okay, so you have your liver, spleen, bone marrow, and lymphoid organs. And even from these four, the most important one you have to remember is the liver. Okay, and this is a side-by-side comparison of all three, because I know if you see it slide by slide, because you see it slide by slide, you still won't be able to get a difference out of it. But this should help you guys. Okay. You can see the continuous capillary. It's it has a defined structure and a thick wall. It's like a proper wall, and it has parasites in it. Fenestrated capillaries, the wall, it's not a proper wall. Okay. The wall is just made up of some sand and cement. Okay. You can say just an analogy. Okay. Continuous capillaries, you have a solid wall with parasites. Fenestrate capillaries, the wall is not solid, it's just an old wall of sand and cement, you can say, as an analogy, okay? And your sinusoidal capillary, there is no wall. It's just packs of cement and sand lying here and there, okay? In second. See this, it's like a proper defined wall, okay? Uniform, proper, defined, and obviously you have the parasites. Fenestrated, the walls are, they're not defined, they're kind of porous, okay? And for as for sinusoidal, uh, it's not even a proper wall, it's just breakage, you know. So you really have no structure over here. This lack of structure allows for greater permeability you see in sinusoidal capillaries. Okay. Oh, yeah. So now we have something called post capillary venule. Okay. Till now we discussed about capillaries. From the capillaries, it goes into the venule. Okay. The capillaries merge into the venule. Okay. And post capillary venule, it just comes after the capillaries. Okay. These are the capillaries, and just after that, you're gonna have a post capillary venule. Now it's unique because first thing, it does not have an analogous artery. Okay. Arteriole, many people might think that arteriole is an analogy to the post capillary venule, but by function, it really is. Okay. It's unique in a way that it's a venule by location. Yes, it comes after the capillaries, so it's a venule by location. But by morphology, by structure, it's still a capillary because this thing, it has like an endothelium and like basement membrane. So by structure, by and large, it's still a capillary. Okay. It does not have analogous arteries, and its main function is extravasation and diaphoresis. Big words. Let me break it down. Extravasation is basically this process of something leaving the blood vessel. Okay, extra or outside. Vasation, the process of leaving the blood vessel. Okay, diaphoresis, it's just uh, almost the same name for it. Okay, so now we'll be talking about migration of cells from blood to the extracellular fluid. So cells actually can leave the blood and can go into the extracellular fluid outside the blood. So outside even the tunica externa, okay? Outside the tunica externa in the extracellular fluid or the extracellular regions where you have different kind of blood vessels. Sorry, the different kind of cells, okay? So for example, you have, let's say inflammation, let's say bacterial invasion or an injury or something like that, okay? What happens is to combat this injury, to combat the infection, you're gonna have neutrophils. Neutrophils are gonna move from the blood vessel into the extracellular fluid. Okay. Now, this movement of neutrophils is by three steps. Okay. And to, just remember this word add. Okay. Each letter corresponds to one step. MAD. Okay. First step is migration. So cells move from the center of lumen. So we have this lumen, and the cells are usually at the center. Cells flow at the center. Okay. And from there, now they move towards the wall of the blood vessel. So from the center to the wall. So that's migration or margination. The, for example, let's say neutrophils. Let's take neutrophils for an example. They move from the lumen and they go to the margin of the post capillary venule. Okay, that's the, now once it moves there, the neutrophil attaches itself on the wall of the post capillary venule. Okay. Now this attachment is done by something called p-selectin adhesion proteins, which are produced by the endothelium. Okay. Remember this. Remember this. Remember this. Remember this. Okay. P-selectin adhesion proteins are an important thing which help in attachment of the neutrophils or some cells onto the, what do you call it? onto the postcapillary venule. And diaperesis is basically the cells exiting the postcapillary venule and moving into the surrounding areas. So they just open the door and move. Okay, that's the thing. Now, all this movement of such huge cells is possible due to absence of intracellular tight junction complexes. Okay, so how we have like gap junctions, we have tight junctions and all that, right? So post capillary venules, they don't have tight junctions. They don't have tight junctions. Okay, so since they don't have tight junctions, they're pretty loosely organized, and cells can just go through and identify these. We have two structures here. I'll give you guys a hint. Both of them are different histology slides of the same structure. Same structure. Okay. Can anyone identify them? It was not discussed till now. Okay. But if you can figure out the characteristics of surrounding tissues, I think you will be able to figure it out. Okay. Yeah, you can unmute the mic and say it too. And if you guys don't know, just say it in the chat. It's okay if you guys don't. I'll explain why. Left is post capillary venule. Yes, left is post capillary venule. Okay. And I gave you guys a hint right now. Okay. Both of them are of the same structure. And someone guessed it as the post capillary venule. And yes, they're right. They're right. Okay. It is the post capillary venule. So, how is it a PCV? Okay. The central region is the blood. The blue cells in the surrounding you can see are actually the white blood cells which are migrating out of the cell. Okay. Now, here, the structure which was here under the square right here, that was the PCV. Okay. If you get this image in the exam per se, if you want to recognize how that's a PCV, so appreciate the presence of surrounding capillaries. So just around the post capillary venule. See, as I told you, there's capillaries. From there, you're gonna have the post capillary. So again, here you have capillaries pretty close to the post capillary venules. Okay, and something bigger than this is the muscular venule. Checkpoint. So congratulations, we are done with uh, cardiovascular histology. Okay, you guys have any questions so far about this lecture or even the first two lectures? Say, okay, we'll be moving on to pulmonary histology from now. Do you guys have any questions? No questions at all? You can unmute the mic and ask. Seems no one has any questions, so let's move. Let's now start with pulmonary histology and let's start with the pulmonary basics. Okay. Structural organization of the respiratory system. So we can organize the respiratory system. First of all, let me tell you what the respiratory system is. So respiratory system, in simple words, is basically the system which helps your body to replenish the oxygen stores in it. Okay, so you inhale air, air has oxygen. Respiration happens where you exchange the oxygen from the surroundings into our body, and now our body is restocked with oxygen. Okay, so that's what respiratory system is. It helps in this process. Now we have two bases of organization of the respiratory system. Okay, one is the upper respiratory tract, and the other is the lower respiratory tract. Okay, the upper respiratory tract structurally it has the nasal cavity, the paranasal sinuses, and the pharynx. The lower respiratory tract is going to have the larynx, the trachea, bronchi, the divisions, and the lung. Okay. Now, this organization is based on their embryological origins, and we really don't have to know all that. Okay. That's the structural organization. Functional organization is organizing the structures based on their function. And we usually use the functional organization when we talk about the respiratory system because you have a clear cut division of what does what. Okay. The conducting portion, it just moves air from the outside. into the body and cleans it. And the respiratory portion, that's where the gaseous exchange happens. Okay. So you can see the diagram here. You can see the diagram here. You have the trachea. Let's start here. The trachea, the primary bronchus, okay, and then you have the secondary bronchi, the lungs, okay. All of this, even up until the terminal bronchioles. You're just moving here, okay. Now, from the respiratory bronchial onwards, some gas exchange starts happening, okay. So that was all for the basics. And let's start with the second last lecture we have, respiratory tract histology 1. Okay, I'll be discussing more about the conducting portion here, and we'll start with the nasal cavities. Okay, the nasal cavity is this air chamber. You can see it right in your nose, right here. Okay, this is the nasal septum, and the chambers inside. Those are your nasal cavities. Okay, and the nasal chambers actually have three regions: the nasal vestibule, the respiratory region, and the olfactory region. Okay, you can see the diagram here once again. The vestibule is this region. It's like the outer region. Okay. So when you look into your nostrils, okay, like when you look into your nostrils, you just see the vestibule. Most probably you're just gonna see the vestibule region. Okay. More deep to it, you're gonna have this respiratory region. It's much bigger and it's deep into our heads. Okay. So we can't see it. We can't really see it. Okay. It's deep into our heads. Olfactory region, we are not seeing it. It's even deeper than the respiratory region. Okay. So if we like look into our nostrils, you get to see the vestibule. Okay. And the nasal septum is just this thing. Okay. So, so first we'll be talking about the nasal vestibule. Okay. The outermost part. This is the outer part of the nose, and it's just around the nostrils. Okay. The epithelium here, since it's outer structure, it's more close to your skin. It's gonna be like the skin, which is stratified squamous epithelium at first. Okay, it's gonna be at first stratified squamous epithelium, but as it approaches the respiratory region, it transitions into pseudo stratified colon. Okay, but most of the nasal vestibule is in fact stratified squamous epithelium. Okay, so this stratified squamous epithelium, that's the main epithelium in the nasal vestibule. The transition that occurs later. Okay. Now you have other structures like the sweat and the sebaceous glands and special coarse moist hair called the vibrissae. Okay. Now people have like hairs in their noses. Okay, it's common to see hair in the nose. That hair is called vibrissae. Okay. There was no previous year question asked on this, but let it be. Okay, whenever you see like a weird name like this, a new name, it's good to memorize that because chances are they will ask questions from you. Chances, okay? Yeah, so the hairs in your nostrils, those are vibrissae. So done. Let me see this thing. Okay, these are your sebaceous glands with your SNI in it. These are your vibrissae follicles. Okay, right over here. These are sebaceous glands. Like this is the skin, like the outer part of the nose. Okay, so you're done with that. And let's go to the next one. The respiratory region. Okay, we are done with the nasal vestibule, and now we are talking about the respiratory region. Now, the respiratory region it starts getting a bit interesting. Okay, how? First of all, is the largest part of the three, and it's lined by something called the respiratory mucosa. Okay, the respiratory mucosa it has an epithelium and something called a lamina propria. Okay, did we have a lamina propria so far? I don't think we had something called a lamina propria so far. Okay, so the lamina propria, this thing, on top of this, you're gonna have the epithelium. So it's epithelium, basement membrane, and then the lamina propria. Okay. This is the largest part, as I told before. Second part. Okay, we can look at the diagram here. It's the vestibule, and this big part over here, that's the nasal mucosa, and its medial wall has the septum. Medial is more towards the more towards the central line. Lateral is more towards this line. So your nose, the respiratory region. Okay, if you move medial, you're gonna approach the septum. This thing. Okay, and more towards the lateral sides, you're gonna have the nasal concha. Okay, these are bony projections which come from bendings in the nasal bone over there. Okay, it's not really important for you guys to know it from histology. You guys took it with anatomy. I'm pretty sure you guys took this in anatomy. Was good. Next one, respiratory region. Now we'll talk about the epithelium, and then we'll talk about the lamina propria. Okay, epithelium. Remember this. Remember this. Remember this. Pseudostratified columnar epithelium is the same thing as respiratory epithelium. They are used interchangeably because later down slides I might change the names. Okay, they're the same thing, just they have different names. Okay, so we have pseudostratified columnar epithelium being the same thing as the respiratory epithelium. Remember this. So in the epithelium of the respiratory region of your nasal cavity, you have five kinds of cells. The most abundant one is your ciliated cell. It's your common pseudostratified columnar. This one. This one right here. Okay. You can see it's these are column-like cells with cilia on them. Okay. So these are actually ciliated pseudostratified columnar. Okay, I forgot to mention that. I think with the ciliated. Okay. Because that's where you're gonna have the mucociliary escalator to sweep away mucus. Okay. So these ciliated cells, they contribute to the mucociliary escalator. Okay. And this escalator basically it's like a bunch of cilia lined with each other. They they sweep in one direction to move mucus. Okay. Goblet cells. Goblet cells are these things right here. You can see this. Okay. The unique thing about goblet cells is that the nucleus, it's at the base, it's pressed at the base, okay? And the apical part, it has these granules, which is filled with mucus. Okay, so if you guys would, guess the function of the cell, its function is producing mucus. Remember this, goblet cells produce mucus. Goblet cells produce mucus. Remember, okay? Just keep it in mind. So you can see this over here, these small bead-like granules over there. Okay, so that's your what do you call mucin granules. Inside of them is mucus, and they're gonna get released outside. Okay, now we have brush cells. Brush cells are these cells right here. They don't have long cilia. They have microvilli, and they have some nerve endings over here. Okay, they should have nerve endings over here, and they're chemosensory receptor cells. The function really is not important here. Basal cell, okay? The second cell over here. Okay, this is the third cell. Basal cell, this is the fourth cell. These cells, they actually don't cover the thickness of the epithelium. They don't cover the thickness of the epithelium. Okay, they're just at base, and basal cells, their function is one. It's an important function. Stem cells. So they work as stem cells, and basal cells are capable of generation of ciliated, goblet, brush, and small granule cells. Okay, they are stem cells for the epithelium. And finally, we have something called your small granule cells. Again, whenever you see a unique name, it's best for you guys to memorize it. Small granule cells are more popularly known as Kulczycki cells. Kulczycki cells. Okay. Now these resemble basal cells in the fact that they do not cover the thickness of the epithelium, but they also contain small granules. So that's why they're also called small granule cells. Okay. And their function is that they are the endocrine cells of a system within your nose. This system is called the diffuse neuroendocrine system. Now, the details on this system, you guys don't have to know. I don't know. Okay. Just know that these are endocrine cells for the system. That's all you got to do. Okay. Here you go. These are some more images to help you understand the concept. Okay. So these are more clear, I would say. This is a goblet cell because of the mucin granules. So whenever, guys, keep this in your brain. Whenever you want to figure out which cell is a goblet cell, search for the mucin granules, these huge granules, and the nucleus is going to be probably at the base. It's going to be one cell with the nucleus at the base and the granules being on top. Okay. Breast cell, if you want to identify breast cell, just know that it has like an afferent nerve connecting the breast cell. Okay. Basal cells, they're pretty down. Okay. Ciliated cell, these are the common cells you have. Okay. All the cilia you see, that's because of these ciliated cells. They cover the entire span of the epithelium. Okay. And this is another image to help you guys understand. So, so we are done with the epithelium. Now we'll talk about the lamina propria. Okay. The lamina propria, it has loose connective tissue, it has blood vessels in it, and these blood vessels, they have blood to provide with warmth and humidity. Okay. Now they have mucus glands. The mucus glands over here, uh, with serous demilunes. Okay. Now what's all this? Mucus glands, serous demilunes, all that. See, one second. Just look at the image. See this thing? That's gonna be a mucus gland. Okay. Now this produces mucus. Secretion is the mucoid secretion. But there will be some cells within this gland which also produce serous secretion. Now these cells are crescent shaped. Now you don't really have to know that in context of the respiratory region. Okay, these cells, they have a serous secretion. So these are called serous demilunes. And function of the respiratory region, the overall function of the respiratory region is whenever you breathe in air, the respiratory region conditions it, makes it warm and clears it. Okay, makes it warm and makes the air clear. So we're done with that. Okay, luceti, capillaries, and venous plexuses for warmth and humidity. Mucus glands with serous demilunes function conditioning of intake and air, making it warm, moistening it, and filtering it, and providing an immune response. Okay, nothing to explain here. Now we have the olfactory epithelium. Now this epithelium is the one which is like the most back. So we had the nasal vestibule, we had the respiratory region, and then we had the olfactory region. Okay, is the uppermost part. Okay, it has the olfactory mucosa. Mucosa. Whenever there is mucosa, whenever you guys hear mucosa, remember this. Mucosa equals to epithelium plus lamina propria. Remember this all the time because you're gonna hear the word mucosa a lot in respiratory histology. Okay, so mucosa, yeah, epithelium plus lamina propria equals to mucosa. Remember this. And the most important function of the olfactory epithelium is detecting odor. So whenever you smell something good or smell something bad, it's because of the olfactory epithelium doing its job. Okay? So the epithelium is again pseudostratified columnar, and it has four types of cells. The the two most important ones are your olfactory receptor cells. These are bipolar cells, and they go through the full thickness of the epithelium. Okay? So these cells are right here. Okay, these are basically bipolar neurons. These are basically neurons. Okay, they have a thickness of epithelium. They go to the CNS. You can see this thing, right? This nerve going towards the CNS. Okay, and they are the main cells in detecting smell. Then you have something called supporting cells. Okay, this right here, just surrounding the olfactory receptor cell, you have the supporting cells, and it provides support to the function cells. So, for example, you have some particles which have some smell. Let's say, okay. what happens is these supporting cells, these produce some proteins which bind onto the odorant. Okay, odorant is the thing which is causing the smell, the steam, smoke, whatever, the particles which cause the smell. So those are odorant. So the supporting cells, they produce odorant binding proteins, and this helps uh, in the detection scent from the olfactory receptors. And basically, you don't have to remember all that. Just remember, guys, just remember that supporting cells support the olfactory receptor cells. That's it. Basal cells, again, wherever you see basal cells, those are your stem cells. And again, just like your respiratory region, they don't cover the thickness of the epithelium. They're just at the base. Okay. Then you have burst cells. Nothing important. Okay. Cool. The lamina propria. Okay, again, it's the same thing: loose connective tissue, blood vessels, olfactory nerves, and you have your olfactory glands known as the Bowman's glands. Okay, olfactory glands, the Bowman's glands, they produce this watery substance which help detection by the olfactory cell cilia. Okay, so basically, again, olfactory glands, their work is similar to supportive cells. Just know that. Okay, so you have your paranasal sinuses. Paranasal, para means a bit outside, nasal of the nose. Okay. So this is this right here, this is your nose. Okay. And surrounding it, right around here. Okay. Right around here. That's where your sinuses lie. We have four sinuses: frontal, ethmoidal, sphenoid, and maxillary. Okay. I think you guys took this in anatomy too, the functions and the details of them. Just know about the four sinuses: frontal, ethmoidal, sphenoid, and maxillary. They are lined with respiratory epithelium, which is also known as a pseudo-stratified columnar epithelium. Okay, so wherever you see respiratory epithelium, it's the same as pseudo-stratified columnar epithelium, and the function is to decrease weight and add resonance to voice. Okay, so now we are done with nasal cavities. That was a big one. Pharynx is going to be a bit small. Pharynx, you have it's a connection between your nasal cavity, oral cavity, and the what do you call the esophagus and the trachea. It's like the general region over there. Okay, once I'll show you the image. So this is your nasal cavity. Okay, this is your oral cavity. This is your esophagus, and this region right here, that's gonna be your pharynx. Pharynx is the common point of like the common meeting point of everything. Okay, so it's divided into three parts, and if you remember the names right here, if you remember the names, that should be enough for you guys to remember the functions too. Okay, so the pharynx overall is divided into three parts. We have the nasopharynx, just by the name, nasopharynx. So it's behind the nasal cavity. Oropharynx is behind the oral cavity. Laryngopharynx is behind the laryngeal cavity. This is not laryngeal cavity, wait, it's behind the larynx. Okay, and the nasopharynx, it has respiratory epithelium. Oropharynx and laryngopharynx, they have stratified squamous epithelium. Just know that. That's it. Okay. Also, the lamina propria of the nasopharynx. Okay, just the upper part. Okay, it has something called the pharyngeal tonsil. Okay, so this thing, this is the nasopharynx. This has the pharyngeal tonsil right here. Also, an important thing I forgot to mention, remember it, is that the eustachian tubes from the ear, they open in the nasopharynx. Okay, eustachian tubes open in the nasopharynx. Remember that. I forgot to mention. Just remember about the eustachian tubes. Okay, because they did not ask anything from that as far as I remember. Just remember it. It's not that. It's not high. Okay, so now we are moving on to larynx. The larynx. The larynx. Okay. So we talked about the pharynx. Second. So far, we talked about the pharynx, okay, and how the pharynx is like a common meeting point of the nasal cavity, the oral cavity, and the esophagus and trachea and all that. Okay, the larynx. Okay, see, you have the pharynx, and then you have the trachea here. Okay, you guys can see, right? So you have the pharynx here and the trachea here, and this connection between them. Okay, this connection between them, that's gonna be your larynx. So the pharynx take two parts again, and the laryngopharynx. So it continues as the epiglottis down, or it continues as the larynx in the front. So epiglottis is from the back. Okay, so more towards your back, you're gonna have your epiglottis, and more towards your front, you're gonna have your larynx. Okay, see the image over here. This is the continuation towards epiglottis. Okay, this was your pharynx. Okay, and see how it's branching off this midway. Okay, this midway connection. That's where your larynx is. Okay, that's the larynx. So the larynx is basically a connection between the pharynx and the trachea. Just remember that. Okay. Now, as for structure, it's formed by irregular hyaline and elastic cartilages. Okay. I gave some names, but they are not really important. You guys covered that anatomy. Okay. Function is phonation. So the larynx, you're gonna have the vocal cords in the larynx, which helps you make sound. Okay. You can talk because of the vocal cords in the larynx. Okay. Uh, So epiglottis. Okay. We had the pharynx. Okay. We had the larynx and we had the epiglottis. Okay. The epiglottis basically covers the larynx. Okay. This is the top part of the larynx and it's flat and it's made up of elastic cartilage. Okay. This is an important point. It's flattened and it's made of elastic cartilage. Okay. It's an important. And it has two faces. This is another important point. And let me tell you how important this point is. Okay. Three times. Three times in the past have questions been asked from this, the lingual and laryngeal phases. Okay. I guess even more than three. This is really so the epiglottis having the lingual phase and the laryngeal phase. The lingual phase, just by the name, is towards the tongue, and the laryngeal phase is towards the larynx. Okay. Lingual phase, just like the tongue, it has stratified squamous epithelium and laryngeal. Okay. It starts as stratified squamous epithelium, but quickly transitions into respiratory epithelium. I'll tell you how. This was the lingual surface. Follow the cursor. Okay. This was the lingual surface. So all of this, we are still at the stratified squamous epithelium. Okay. This is where the transition happens. And from here, we are now entering into the respiratory epithelium. Okay. Mixed serous or mucus acini. So you're gonna have some um, mucus and serous glands. It's not really that important. Okay. And from this slide, if you would forget everything, just remember that the lingual surface has stratified squamous epithelium. The laryngeal surface has mainly respiratory epithelium. One question, I'm 60% sure one question is going to come from here. Okay. So these are just some more histology slides. Okay. This is the this is the glottis. Okay. You can see like it's like a flap-like structure. Okay. It's a flap-like structure which covers over the larynx when you eat food. Okay. Because you don't want the food to enter the trachea. So the epiglottis covers the larynx. So the food can pass into the esophagus. Okay. So if we talk about the lingual surface, follow this black box right here. This is the black box. Okay. Stratified squamous epithelium. Then we'll come to this side. Okay. The red box. Transition. So this right here was stratified squamous epithelium. Now this is the point of transition, and here you have respiratory epithelium. Okay. Obviously, it contains a respiratory epithelium. You can see it's this elongated epithelium. Okay. And if you carefully see, you have some cilia on here, okay? And it looks stratified, but it's actually not stratified. It's pseudo stratified, okay? This is properly stratified. You can see layers on layers on layers over here. It's stratified with epithelium. Okay? That was it for epiglottis, I guess. Now we'll talk about the vocal cords, okay? The vocal cords are your actual organ of phonation, which means you can produce sound because of your vocal cords, and they lie between the epiglottis and the trachea, midway in the larynx. Okay, and get this image over here. So this right here is a false vocal cord. This is a true vocal cord. Okay, and the gap between the false and the true vocal cords right here, it's called the vestibule. Okay, this false vocal cords is the upper pair, and they are covered in respiratory epithelium. True vocal cords is the lower pair. Now, this is the structure, like the histology slide of the true vocal cord, okay? And it actually has non-keratinized stratified squamous epithelium. Okay, remember the non-keratinized part, because this has been asked before. Okay, I'm telling you, the the epithelium on the false and the true vocal cords. This has been a previous year question. I think it was in my exam. Okay, so now you have your respiratory epithelium on your false vocal cords and your true vocal cords. They have your stratified squamous epithelium, which is non-keratinized. Okay, we have something called Ranke space, which is basically the lamina propria over here. Okay, in the true vocal cords, the lamina propria, it's called the Ranke space. Important. This is also a previously asked question about the Ranke space. Okay, it has been asked before. Again, as I said, you guys, for like the fifth time, whenever you see like a new name or like a person's name, remember it. Okay, vocal ligament is this thing. Okay, it's under the epithelium, under the lamina propria, aka the Ranke space. Okay, and it has dense regular elastic connective tissue. Okay, now an important structure in the vocal cords is the vocalis muscle. The vocalis muscle is also known as the thyroarytenoid muscle. Okay, and this thing is responsible for contracting and maintaining tension within the vocal cords. And this is why you can create different frequencies of the voice. Depending on how contracted or relaxed the vocalis muscle is, you can create different frequencies of voices. Okay. So the most important things I would like you guys to remember from this slide is the epithelium of the false and the true vocal cords and the vocalis muscle. Okay. Again, epithelium of the false vocal cord being the respiratory epithelium, the true vocal cords being non-keratinized, stratified squamous epithelium, and you're gonna have your vocalis muscle, also known as the thyroarytenoid muscle, which is responsible for regulating tension and producing sounds of different frequencies. Okay, this is just another image. Uh, you guys can see this. You have your respiratory epithelium on the false vocal cord. Okay, the blue box right here. It has the respiratory epithelium for the false vocal cord, and the green box, it just shows some glands. Okay. Now your true vocal cords, they have stratified squamous epithelium. Look at the black box over here. Okay. And the red box, it has your vocalis muscle. Okay. Uh, Different frequencies of voice and maintaining tension in the vocal cords. Okay. Because if this muscle fails, your vocal cords will not contract and you will not be able to talk at all. Okay. We're done with the nasal cavities. We're done with the pharynx, larynx, and trachea. Now, from the trachea onwards, guys, it's gonna be pretty standard. Okay, it's gonna be easy to track from here. Okay, because first of all, let's talk about the trachea. Trachea. Now we are done with the nasal cavity. We moved into the pharynx. We moved into the larynx, and now we are in the trachea. Okay, it's a flexible tube which like starts from here, and I guess it goes until like here, 10 to 12 centimeters long. Okay, starts at the end of the larynx and ends at a point called carina. Okay. Carina is a point of bifurcation, and what happens here is one second. This point right here, That's your carina. Okay. What happens here is the trachea being one singular tube, it divides into the primary bronchi, the left and the right primary bronchi. Okay. So the point of division, that's the carina. Again, it's a new name. Remember, the trachea, it consists of the mucosa, submucosa, the cartilage layer, and the adventitia. Okay. I mean, I'm going to be explaining you guys about this, so remember it. Okay. The arrangement of the cartilaginous rings. Okay. So the rings in the trachea are basically these C-shaped rings. They're arranged in the C shape. Okay. And they stack on top of each other. In this way, they prevent uh, the trachea's collapse. Okay. The trachea does not collapse within itself, because if it collapses, we can't breathe. And it provides flexibility. Okay. Remember the layers. Okay. Remember the order, how it goes. If we go from the inside to the outside, so from the lumen of the trachea to like the outside tissues. So we start at the lumen, and then we go hit the respiratory epithelium, and then the basement membrane, and then the lamina propria. Okay. So respiratory epithelium, basement membrane, lamina propria, these three are part of the mucosa. Okay. Then you have your elastic membrane and submucosa again being part of the submucosa, and then you have your cartilage layer and then adventitia. Okay. Anytime you see adventitia or externa adventitia, just means it's the outer layer. Okay. So go to the mucosa. Okay. The mucosa, the epithelium again, uh, as we talked before, the epithelium is a respiratory epithelium with a thick basement membrane. The lamina propria has these unique structures called bronchus associated lymphoid tissue. That's for immunity. Okay, just know the name. Know the name. Okay, the lamina propria of the mucosa in the trachea having something called bronchus associated lymphoid tissue. Now, other than that, it's pretty normal. It has the loose connective tissue and all that stuff. Okay, it's very cellular too. And lamina propria almost all the time has some kind of loose connective tissue. It's what basically a lamina propria is, just a bunch of connective tissue. Okay, but in this case, it has something special, and that special is bronchus associated lymphoid tissue, aka BALT. Okay, now we have submucosa. The submucosa lies under the mucosa. Okay, again, this thing has loose connective tissue, but it's a bit denser than your mucosa, and the differentiation. Okay, between the mucosa and the submucosa. It's done by something called an elastic membrane. Okay. Till now we had the basement membrane. Till now it was just the basement membrane. Now we have something called an elastic membrane. The basement membrane is something on which just the top layer of epithelial cells lie on. That's your basement membrane. So you have your basement membrane, then you have your lamina propria, then you have your elastic membrane, which separates the lamina propria from the submucosa. And the submucosa, it's well under the elastic membrane. It consists of large blood vessels, okay, and it has a huge glands. So in this picture, you can see it's this thing labeled glands. Okay, so yeah, that's cartilaginous layer. Cartilaginous layer. Okay, now you have 16 to 20 C-shaped, what do you call, cartilages stacked on top of each other, and they provide flexibility. Okay, and the thing about the cartilages, see, you have your C-shaped trachea. Okay, this is one end of the C shape, and then this is another end. There is a muscle or a structure which connects these two. Okay, that's called your trachealis muscle, and this is a smooth muscle. Along with the trachealis muscle, you also have your fibroelastic sheath, but the most important structure to remember here is the trachealis muscle. That's the important structure over here. And then you also have some seromucous acini, so some glands. Okay, adventitia, the same thing. It just connects the trachea to the outside regions. Okay. Uh, Now you have some diseases. Okay, I think this page marks the end of this lecture. So we have inflammation. So whenever there's inflammation, you add itis at the end. So for example, you have rhinitis. It's just going to be difficult to breathe because your blood vessels have engorged over here. Okay, now you have something called anosmia. Remember, anosmia is what do you call anosmia is inability to breathe or inability to smell, not inability to breathe. I'm sorry. Inability to smell. It happens due to depletion or dysfunction of the supporting cells. Remember, the supporting cells, not the olfactory cells, okay? Because see, virus-induced anosmia is temporary, okay? So in temporary loss of smell, you're gonna have destruction of the supporting cells. If you have destruction of the olfactory cells, that's gonna be a permanent loss of smell. Okay. Now you have Singer's nodule. Look at the image. It's just a benign polyp of stratified squamous epithelium of the true vocal cord. Okay, and it affects your voice. It's just that. Just know about Singer's nodule. So now we another game of identify them. Come on. We have ABC on this side, ABC on this side. Anyone? Anyone? There are even enough. Can anyone find out what A, B, and C are? Okay, so I guess not. We have these were goblet cells. How do we know those are goblet cells? I mean, not breast cells. There were no breast cells. Sorry. A was goblet cells. Okay, because you can see these mucin, these white granules of mucin, right? You see these mucin granules. Okay, uh, that's a goblet cells, and then there are ciliary cells or the ciliated cells, and the basal cells, the ones which are at the bottom. That's the basal cells. Here again, this was your respiratory epithelium. I gave you guys a hint because of basement membrane being there, and then this another membrane being there. Okay, that was the hint over here. So anytime you see this, just know that this is the respiratory epithelium. The respiratory epithelium is separated. The the respiratory epithelium is separated by the basement membrane, the respiratory epithelium, and the lamina propria. And now the lamina propria uh, and the submucous are separated by the elastic. Okay. So here you go. This is the last. We'll start with the last lecture finally. And you guys have any questions so far? Finally, the last one, respiratory tract histology, second part. That's the end point. Okay. Now, so we'll talk about lungs. I don't know why this thing is not going up on this black bar. Okay. This is gonna be, we'll be talking about your lungs. Okay. The lungs, these are spongy air-filled sac covered by a thin tissue layer on the outside called the pleura. Okay. Now each lung has a lobe. Okay. So now we have lungs with these two sacs. Now each sac has some lobes. Okay. So the left sac has two lobes and the right sac has three lobes. Okay. You can see the diagram. Okay, so this is one lung sac, and this is another lung sac. Okay, so the left one has one, two lobes. The right one has one, two, three lobes. Okay, now each of these lobes now have more divisions called the bronchopulmonary segments. So now these are eight on the left side There you go. That's 1, 2, 3, 4, 5, 6, 7, 8, and 10 on the right side. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And these bronchopulmonary segments further divide into hundreds of pulmonary lobules. Okay. Now, these are just tissues. Okay. We see the bronchi penetrating these tissues. Okay. And when you look at the bronchi penetrating these tissues from far apart, it looks like this thing. Okay. It looks like this. So we have the main trunk branching into two primary bronchi. This is again branching into secondary bronchi, again branching into tertiary bronchi, and so on. So it's like a tree. Okay. And this tree is called the bronchial tree. Okay. This tree is called the bronchial tree. The bronchial tree. Look at this time. So we have the trachea, and the point of division at the carina, you have the primary bronchus. The primary bronchus just enters the sac. Okay. The secondary bronchus uh, supplies the lobes. The tertiary bronchus supplies what do you call the bronchopulmonary segments. Okay. And the bronchioles supply a pulmonary lobule. Okay. Just know this. Just know this. Primary bronchus, it does not. Primary bronchus supplies the entire sac. So we have two sacs. Okay. One primary bronchus supplies the entire sac. When it goes inside, it divides into secondary bronchi, which supply, say on the left side, okay, we have two lobes. In one sac, we have two lobes. Okay. So now we have two secondary bronchi supplying each of these two lobes. And then we have eight bronchopulmonary segments. So now we have eight tertiary bronchi supplying each bronchopulmonary segment, and so on. Okay. So now we'll talk about the bronchi, bronchioles, and the terminal bronchioles. Again, if you talk about the wall structure, it's similar to the trachea. Okay. The main thing you're gonna have initially they got the same general structure as the trachea. Later, two major changes happen. The amount of cartilage decreases, amount of smooth muscle relatively increases. Okay, because before in the trachea you had 75% cartilage, right? This was 75% cartilage. Okay, and you had like bit of the trachealis muscle. In bronchi, you will not have a cartilage ring. You'll just have some diffuse, scattered small cartilage plates. Okay. So you can actually see it here. This, okay, this thing, this thing, all of these, okay, they are no longer continuous. Okay, they are no longer prominent and continuous. They're little scattered around, they're like that. And later, uh, yeah, so from the trachea's prominent cartilage, we now have scattered cartilage in the bronchi. But the smooth muscle, instead of being one trachealis muscle, now the smooth muscle surrounds the entire bronchi. Okay, it completes an entire circle. Okay, and becomes more prominent. And as we reach the bronchioles, we skip the cartilage, and all we have is a thick layer of smooth muscle. Okay. So now we got 5 layers here again. Same thing. We got the mucosa. Okay. Here we have the epithelium and the lamina propria. They're the same as the trachea. Muscularis. This is the fifth layer because the trachea had 4 layers: mucosa, submucosa, cartilaginous layer, and adventitia. Okay, those are the 4 layers. Now bronchi we have 5 layers because as I told you guys, mucosa is the same. Smooth muscle, instead of being this one trachealis muscle, what happens is that it now surrounds the whole thing, forming its own layer. Okay, so now under the mucosa, you now have the muscularis layer. Okay, uh, again, as I wrote down here, continuous layer of smooth muscle in larger bronchi. Okay, and in comparison, trachea has a discontinuous trachealis muscle. That's it, just that. That's it. Okay, but in bronchi, you have like a full continuous layer. Now we have what you call it's a mucosa. So we have mucosa, then we have muscularis, then we have submucosa. So people might confuse this. Okay, some people might say submucosa is what do you call it? Four, but no, submucosa is after. So first is mucosa, then it's muscularis, then it's submucosa. And then obviously after that it's gonna be your cartilaginous layer, which has discontinuous hyaline cartilage plates. Okay, and then we have your adventitia. That's the same thing. Okay, now the bronchioles, again, it's almost the same thing as the bronchi. The biggest differentiator, BYQ. This has been a previous year question before. The key differentiator between bronchi and bronchioles is the absence of cartilage plates and glands. By the time you reach the bronchioles, cartilage is completely done for. You just have smooth muscle. Okay, see this thing? Smooth muscle makes a complete round. Okay, and that's your mucosa. Obviously, you can have a mucosa. Mucosa is everywhere. The lamina propria has no glands. Okay. Muscularis is a relatively thick, smooth muscle wall and adventitia. Okay. Now remember this point, guys. Please remember this point. Okay, this has been previous year question. Key differentiator between bronchi and bronchioles is the absence of cartilage and absence of glands. Okay, bronchioles have zero cartilage at all. So now let's start off how the branching goes. Okay, look at this. This is also an important point. It's for you guys to understand. So we have the bronchi, and then we have the bronchioles, the smaller bronchioles, and then we reach the terminal bronchioles. Okay, the terminal bronchioles, they are the last point where you just they're the last point of gas passage. Okay, now respiratory bronchioles, that's the point where you start gas exchange. Okay, and terminal bronchioles are bigger than the respiratory bronchioles. The respiratory bronchioles come after the terminal bronchioles. Please remember this. People get confused here too, just because they think that terminal means the last one, terminal last one, so it has to be the last one. No, after the terminal, you have the respiratory bronchioles. Okay, remember this. Don't get confused. So now we have your epithelium, which lines the bronchioles. The epithelium, which lines the bronchioles, is interesting to say the least. Okay, because since we started off in the trachea, all the way from the trachea to the primary bronchi to the larger to the smaller bronchi, okay, uh, now we are at the bronchioles. All this way, you had just one type of cell in the epithelium. That's your ciliated pseudostratified column. Okay, now you're gonna see changes. Epithelium, which lines the bronchioles, okay, you're gonna have ciliated pseudostratified columnar cells in the larger bronchioles, and once you reach the smaller bronchioles, it's gonna be simple ciliated column, simple ciliated or cuboid. Okay, goblet cells are present only in large bronchioles. They are gone in small bronchioles, and these are important cells. These are very important cells. Again, a previous year question has been asked from you. Okay, club or Clara cells. Again, there is a name here, Clara. That's a new name. Remember it. Okay, club or Clara cells. These are non-ciliated secreting cells. Okay, they secrete surfactant. Okay, they secrete surfactant, and this was the thing on which the previous year question was asked. Okay, so the question went something like, which of the following cells produce surfactant in the terminal bronchioles? As far as I remember, that was the question. And the answer was your Clara cells. Okay. So, yeah, your club Clara cells, they are non-ciliated cells, they are secreting cells, and they secrete two things. They secrete surfactant, which prevents airway collapse in the terminal bronchioles and the respiratory bronchioles. And they have something called CC16. They produce club cell secretory protein, CC16, which is immunomodulatory. That's not important. It's not as important as its function of secreting surfactant. This has been a previous year question. Know this. Club cells produce surfactant in large and like terminal bronchioles. Know this sentence. Club cells produce surfactant in bronchioles. Know it. Okay. And they also act as stem cells for the rest of the epithelial cells. Okay. That's another important function. But the surfactant one is the most important of them. Now, go on to the next one. We are done with the conducting portion so far. Okay. We'll be talking about the respiratory portion. In this portion, you will actually see gas exchange taking place. Okay. So we have our respiratory bronchioles. We are done with the terminal bronchioles. Now we are in the respiratory bronchioles. This also has been a previous year question. Okay. This point, especially, that which structure has both air conduction and gas exchange. Okay. And the answer is respiratory bronchioles. Really important. This has been asked multiple times. Okay. There are transitions. Yeah, because they have both functions. And the wall here has just two components. You don't have your what do you call it? You don't have your adventitia, your cartilage, your all that stuff. Okay, you don't have that. All of that is done for because respiratory bronchioles are just too small for them. Okay, now the wall here has just two components. Yes, simple cuboidal epithelium with club cells. Okay, also alveoli. We are finally reaching the end. It's alveoli. Okay, look at this. Look at the box right here. Okay, this whole thing, this whole thing is a respiratory bronchiole. Okay, this was a terminal bronchiole. The terminal bronchiole, it branched off into the respiratory bronchioles. Okay, and within the respiratory bronchioles, you have your simple cuboidal epithelium. Okay, uh, bit bit of smooth muscle, I would say, and alveoli start here. Okay, so once the respiratory bronchioles terminate, they terminate into these alveolar sacs. Okay, so the walls themselves, they start having alveoli, but at the end, it's not completely alveolar. Okay, so just remember that alveoli, they start appearing at the respiratory bronchioles. Okay, so you can see this is also an alveoli. Okay, these are also smaller alveoli and all that. Okay, the most important point I would like you guys to remember from this slide is that it's the transitional zone. Okay, so it's like a transitional zone. It transitions between both conduction of gas and gaseous exchange. Okay, this is the most important point I want you guys to remember from this slide. And let's go to the next one. Alveolar duct. Okay, so alveolar duct and sac. The thing about this is that it's not really clearly written in the slides, and I tried to make it as simple for you guys. They don't actually have any true walls. Okay, so we are done with the terminal bronchioles. We now go into the respiratory bronchioles, and these end in the alveolar duct and sac. Okay, both of them, both the alveolar duct and the alveolar sac, both of them, they don't have true walls. Okay, it's just that their walls, they are just a bunch of alveoli opening at the peripheral boundary. Okay, so you can see here. Okay, uh, see this. This is supposed to be your alveolar duct. Okay, this thing is supposed to be alveolar duct, but you can see that it's just these alveoli. Okay, on the wall, you just have alveoli on the wall. Okay, you can see it's everywhere. It's just alveoli. Okay, even on alveolar sac, you just have these alveoli. Okay, there is like no true wall over here anymore. It's just alveoli and alveoli. Okay, remember the one thing I want you guys to remember from here is that alveolar duct and sacs, they don't have any true walls, and their walls are just a bunch of alveoli. Okay, so now we're finally at alveoli. This is the terminal airspace for gas exchange. Okay, this is the point where gaseous exchange takes place. Okay, there are a lot, approximately 150 to 250 billion. Okay, and they just contain epithelium. That's it. Epithelium, maybe basement membrane. Okay, epithelium, basement membrane. That's it. You don't have lamina propria. You don't have cartilage. You don't have smooth muscle. None of that. Epithelium, basement membrane, because it's really thin, and you need to make it thin enough for diffusion to occur fast. Because at this point, you're going to have capillaries, which will bring in blood. Okay, and here there's the exchange. Carbon dioxide from the blood goes into the alveoli, and the oxygen in the alveoli moves into the blood. Okay, so once again, look at this. Okay, these are your blood vessels. All of these are your blood vessels. Okay, these circular structures, those are alveoli. See how they're surrounding them. Okay, this is where the exchange happens. And alveoli, they alveoli, they have two types of epithelium. Just to remember these two important, and many questions have been asked on these two epithelial types: type 1 pneumocytes and type 2 pneumocytes. Okay, and let's differentiate between them, because the best way to actually memorize these is just differentiating. Okay, why is this type? So these are actually very different. Type 1 pneumocytes are extremely thin and flat squamous cells, and type 2 pneumocytes, which are also known as septal cells, are more bulgy, they are more round. Okay, type 1 pneumocytes by absolute number, okay, 40. Type type 1 pneumocytes. Type 2, it's 60. Okay, for example, if there are 100 total cells in the epithelium, so 40 of those cells are going to be type 1 cells, 60 of those cells are going to be type 2 cells. But if you're going to see by area, since the type 1 cells are extremely thin and extremely flat, they actually cover 95% of the area of the alveolus. Type 2 pneumocytes cover just 5%. Okay, type 1 pneumocytes, they're not capable of cell division, and type 2 are basically progenitor cells for both type 1 and type 2. Okay, type 1 pneumocytes, main function, exchange of gases. Since they are so thin, diffusion occurs through them easily. Again, remember, they are thin, diffusion occurs easily, so exchange of gases happens here. Okay, type 2 pneumocyte, it produces something called a surfactant, which prevents the alveolus from collapsing when there is no air in there. Okay, the main function of a type 2 pneumocyte is the secretion of surfactant. Main function of type 1 pneumocyte is gas exchange. Okay, obviously it participates in the blood-brain barrier. That's part of the gas exchange. Okay? Now we have surfactant. So again, your surfactant is synthesized in type 2 pneumocytes. Okay, and it's stored in laminar bodies. That part's not really important. Okay, just remember that surfactant is produced by type 2 pneumocytes, and it's a mix of DPPC, neutral lipids, and surfactant proteins. Three things that might come in the SAQ, I would say: DPPC, neutral lipids, surfactant proteins. Remember these three. Now, the full form of DPPC is dipalmitoyl phosphatidyl choline. If you guys want to remember this, remember it. I feel DPPC should do fine. I memorize the name. Okay, dipalmitoyl phosphatidylcholine, DPPC. Okay, then you have neutral lipids and surfactant proteins, and its function is to decrease alveolar surface tension, because if there is surface tension in the alveoli, once there is no more air to keep the alveolus open, the alveolus collapses. So this thing prevents the collapse of alveoli. Okay. Now, what happens if you don't have the surfactant? Your alveoli collapse, and that usually is seen in neonatal respiratory distress syndrome. So, if you have a child who is born preterm, let's say a child is born preterm, term is 9 months. If he's born before, his lungs are not well developed. So, the type 2 pneumocytes are not well developed, and they will not secrete surfactant yet. Okay, so if the child is exposed to the air outside, the alveolus collapses. Okay, the lungs can collapse, and it will lead to something called neonatal respiratory distress syndrome. The two most important things, or the three most important things I would like you guys to remember from this slide, is that surfactant is produced from type 2 pneumocytes. That's one important thing. The other thing is it's a mix of DPPC, neutral lipids, surfactant proteins. Okay, and the third thing is that it decreases surface tension. And if a child does not have it, he can have neonatal respiratory distress syndrome. The name of this disease is important. Okay, neonatal respiratory distress syndrome. This is probably the only clinical case, I would say, out of everything we have learned so far, is important in context of respiratory histology. Okay, now we have macrophages in the lungs. So macrophages are present over there just as like a cleanup. Okay, so if you have an infection in the lungs, if you have some debris in the lungs, So macrophages are there to clean it up. So we have two types of macrophages. One is alveolar macrophage, and the other is a septal macrophage. Okay. So we have the alveolus. The alveolar macrophage is within the lumen of the alveoli. Okay. Septal macrophages are between alveoli. Okay. So these are two alveoli. They have a septum between them. Okay. This gap, this wall, you could say a common wall. That's a septum. So some macrophages lie in there. Alveolar macrophages, they mainly take up dust particles when you inhale air, for say, you have some dust particles. So they take up dust particles, and those macrophages are called dust cells. Another important clinical case, I would say, is if you have heart attack, what happens is blood moves into the alveoli. Okay. And because of that, these macrophages actually take up red blood cells, which have moved within the alveoli. They take it up. So now they are actually called heart failure cells because these macrophages are seen during heart failure. Okay. Now, septal macrophages, they are found lodged inside the intraalveolar septa, and they can actually stay there for uh, for life, basically. Okay. So let me mark it over here. So remember this image. They might ask you something. So this is the lumen. Okay. This is the lumen right here. This is another lumen right here. Okay. And this is the septum. It has some blood vessel in it. Okay. Now, if I talk about, okay, these macrophages, these free cells in the lumen, those are your alveolar macrophages. And now within the intraalveolar septum, you have these black structures. Okay. These are your septal macrophages. Now, why they're so black? It's because they have been collecting dust and debris and they just stay there. As I told you, they live there for the rest of their life. So that's why they're black in color. Okay. That's it. Intraalveolar septum. Again, it's a thin connective tissue with elastic fibers between adjacent alveoli. Okay. And these elastic fibers are responsible for like to stretch the alveoli because the alveoli actually don't remain of the same size. The alveoli, when you inhale a lot of air, they expand, they become big, like a football. When you put air in the football, it becomes big. So this elasticity comes from the elastic fibers in the intraalveolar septum. Okay. And this region is also where the air blood barrier lies. Okay. This is where your blood vessels are. And this is where, what do you call it? The exchange happens. So basically, oxygen from alveolus crosses bunch of layers and goes into the blood vessel. And this blood vessel is present in the intraalveolar septum. Also, we have something called alveolar pores of Kohn, which are basically like a access way. Okay, so we have the septum, like a solid septum, but then there is a hole at the center. Okay, now this hole is going to be an access way to connect various alveoli, and this hole is called the alveolar pore of Kohn. Again, alveolar pore of Kohn. Kohn is a new name. Remember. Okay. Now you have your air blood barrier. Okay, just by the name, it refers to like a bunch of layers through which the gas has to diffuse. It has two portions, thick and thin. Now the thin portion, whenever you think of the air blood barrier, the thin portion is the one which comes into mind because this thing is through which this barrier is through which gases have to pass through. Okay, so the thin portion, the structure itself. So we have first the surfactant. Okay, we have like the alveolar wall, right? So first we have the surfactant. Let me just try. First we have the surfactant, okay, and then we have our type 1 cell. We have our type 1 pneumocyte, and then we have the basement membrane of your type 1 pneumocyte, and then actually after this you actually have the capillary basement membrane, and most often these two basement membranes are fused on each other. Okay, and after this you have your capillary endothelium. Okay, this is how it goes, and function is gas exchange. Now the thick portion, it's very similar in structure to the thin portion, and it has surfactant, type 1 cell, the two basement membranes, and the capillary endothelium. The only difference in the thick portion is that between the basement membranes, you actually have macrophages and fibroblasts. Okay, so the septal macrophages lie here in the thick portion. Okay, septal macrophages lie in the thick portion. That's what you have to. Okay? So we're done. Uh, some diagrams. You can see this thing, right? So it has this surfactant, and then you have a sorry, one second. Yeah. So here's the surfactant. Here's the pneumocyte type 2. This is your basement membrane. This is your endothelium. This is your lumen, and this is your red blood cell. Okay. The same thing over here. It's just more. It's easier to understand. Let's say. Okay. So pleura. You know how I was talking about the heart, and the heart had like this pericardial layer on it to protect it and to, you know, keep it in place. Okay. Pleura is the same thing for the lungs. Same thing for the lungs. It's again a serous membrane with two sublayers, the parietal pleura and the visceral pleura. So again, the visceral pleura is attached onto the lung surface. There is the pleural fluid, the pleural gap with pleural fluid, and then you have your parietal pleura, which lines the thoracic wall. Okay, so parietal pleura is like the place where the lungs attach. Okay, again, just like the pericardia, just like the visceral pericardium, the visceral pleura has mesothelial cells which release pleural fluid into the pleural cavity for lubrication, just like the visceral pericardium. Okay. Now the thing is, both of these layers, the parietal pleura and the visceral pleura, both of these actually meet. Everywhere else they are separate, but later, like these two layers actually, these are separate in this way. Okay, they're separate, and then they meet at the hilum of the lung. Now, the hilum of the lung is the point where actually the primary bronchus enters the lung sac. Okay, that's the point. So that's all it. Oh yeah, just remember that pleura, it holds really well in analogy to the pericardium. So the parietal pericardium, the visceral pericardium, it's analogous to the parietal pleura, the visceral pleura. Same structure. Okay, not the same structure, the same hierarchy and uh, the same function basically. Even here, you have pleural fluid so that when the lungs expand, they don't get damaged because of friction. Okay, again, the same thing to protect from friction. So the blood supply, innervation, and lymphatic drainage. Remember that blood supply in lungs is unique. One blood supply is the pulmonary circulation. The pulmonary circulation is just for gas exchange to recharge the blood with oxygen. And the bronchial circulation, this actually provides the what do you call, the blood vessels with supply nutrients to keep the lung tissue alive. Okay, so the bronchial circulation is basically what every other part of our body has. For example, blood vessels to our muscles is to keep them alive. Bronchial circulation to the lungs is to keep the lungs alive. Pulmonary circulation is for the gaseous exchange, the special function of the lung. Nerve supply is autonomic. Lymphatic drainage is dual drainage. Okay. Now, some diseases we have asthma, we have emphysema, and cystic fibrosis. Now, asthma is first of all, it's a chronic disease, it's an inflammatory disease, and that happens because of overactive goblet cells, increased number of goblet cells. So you now have a lot of mucus, okay? And the muscle layer in here is hyperplastic and hyperactive. So, what do you call the lumen diameter, the lumen of the bronchiole, the lumen of the bronchiole, its diameter is decreased. So that causes asthma. Now, emphysema. Out of these three which I've written here, emphysema is probably the most important because I think this was discussed a lot in physiology. That is because of smoking, what happens is your alveolar walls, they are destroyed. Okay, that causes shortness of breath. Cystic fibrosis is again a genetic disorder where excessively thick mucus is being produced, so that causes excessively thick mucus is being produced, so that causes failure of the mucociliary sclera. It's like, see, if the mucus is thin, your mucociliary sclera, the cilia can effectively brush it in some direction. Okay, but if the mucus is really thick, it's almost solid, the cilia are stuck. They're not doing anything. They try to move, but they're stuck. So this mucus accumulating in one place also has a danger of stuff accumulating on top of the mucus. So you have dust particles accumulating on top of the mucus. You have bacteria accumulating on top of the mucus, and this causes infections. Okay. So identify them is the last round. Okay. Identify them. Identify what's here. So yeah, this was bronchiole and this was like a small bronchiole. So that's walled and this is a small bronchiole. Okay. Let it be. Now, do you guys have any questions? We are done. We are actually done with the 5 lectures. We are actually done with the, yeah, small bronchiole, right? We are done with the 5 lectures. Uh, like any questions? Any questions in all the 5 lectures? Because after this, I'll be solving some questions which have been repeated previously. It's gonna take 10, 15 minutes long. No more questions? Guys, no more questions?
No.
I, I would assume no. So let's start questions. Let's solve some questions, okay? So the cells which aid in surfactant production is present in which of the following? We just discussed it. Just type the answers in the chat, okay? What is the answer for first and the second? Giving you guys 5 seconds. Now the second one, what layers does oxygen have to move through to reach the capillaries? Okay. Read the options that is there. Okay. Okay. So that's right. That's right. The first one, the cells which aid in surfactant production, club cells or Clara cells. Okay. They're present in which of the following? So respiratory bronchi. We took that. Okay. The Clara cells providing surfactant in the respiratory bronchi. This These are previous year questions, by the way, all of these. Okay. Now, which layers does oxygen have to move through? Now, this question, not only was it a previous year question, but it has been repeated in the past years. Okay. So now the answer was yeah. So it goes through the type 1 pneumocyte and then through the fused basement membrane and then the capillary endothelium. The next one. Which artery is shown here? Now, how will a pathologist know he's viewing a bronchiole and not a bronchus? Okay, good, good. Okay. So, yeah, PC4A. Yeah, correct. So the artery shown here is aorta is because you can see a lot of black lines intertwining with the pink lines. So it's a lot of elastic fibers within a lot of what do you call smooth muscle cells. Okay, and how will a pathologist know he's viewing a bronchiole and not a bronchus? This point has been repeated in previous year questions. Absence of cartilage. Okay, bronchi have cartilage, bronchioles don't. Remember this. Okay. Now, what makes the epithelium of the upper surface of the epiglottis? As I told you guys before, the epithelium on the epiglottis was repeated a lot. Okay. So what makes the epithelium of the upper surface of the epiglottis? Okay. And which of the following has smooth muscles? Okay. Which of the following has smooth muscles? It's a good question. I'll give you a hint. The answer is not none of these. Okay. Okay, so someone answered B. That was right. For the fifth one, the answer is B, stratified squamous epithelium. The upper surface of the epiglottis has stratified squamous epithelium, and the lower surface, which is also known as the laryngeal surface, has respiratory epithelium. And as for smooth muscles, myocardium has cardiac muscles. Tunica externa, it has no muscles. It just has collagen. Tunica media has muscles. Okay. Now, what is the direction of ciliary movement in the mucociliary escalator? Seventh question, 7 seconds. Come on. Okay. And for the eighth question, it's a good question. I'll give you 10 seconds for this actually. Okay. Child was born with her heart valves not being able to withstand pressure. So there is some problem with the valves. Okay. Which of the following structures are most likely missing in the wall? This was actually a repeated question again. Okay. All these questions actually repeated. So yeah. Okay. 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 So the answer for the seventh one is A, upwards towards the pharynx. That's why whenever you are cold or you excrete, you cough out mucus or you use a tissue paper to like clean off mucus because the movement is towards your pharynx and then towards your nasal cavity. Okay, it does not go down. It's not in any direction, and D is obviously wrong. Okay, on the eighth one, this was repeated actually about the fibroelastic sheath. As I told you guys, the fibroelastic sheath was pretty important. It's a pretty important concept. It has been repeated before, and a question might come from it. Okay, because since the fibroelastic sheath is not present, the valves are not being sturdy. Okay, they're not being strong, so they're just flapping out. Okay, that's about them. Which type of epithelium do true vocal cords have? Again, true vocal cords, false vocal cords, and their epitheliums. Important, important, important. Okay, 5 seconds for this. Okay. And for the 10th question, which of the following structures should one focus on when studying about dysfunctions in microcirculation? This question is basically asking which structure is the most important one in microcirculation. Again, 5 questions for this, 5 seconds for this. Okay. So ninth one, the answer was A. Again, I told you true vocal cords, non-keratinized, non-keratinized, stratified squamous epithelium. Please do not answer this keratinized, okay? You will get confused. Non-keratinized. And the 10th one, pericytes. Pericytes are the most important cells in microcirculation. That, yeah, because capillaries usually have pericytes. So that's that. Which of the following structures provides surfactant-like material in the terminal bronchiole? Okay, 5 seconds for this. Okay. And this was also a repeated question, as I told you guys before. Where is the Rankin space situated? 5 seconds for this. Come on, guys. These are easy. Come. Okay. Right. So yeah, you're right. For the 11th one, is Clarissons. And for the 12th question, between the two, within the true vocal folds. The Rankin space is again, it's basically the lamina propria of the vocal fold, the true vocal fold. Okay, so it's within the vocal folds. Okay. Now, the 13th question. Failure of which of the following structures leads to mucus movement. Okay, which means mucus is not moving anywhere and it's not blocking the air per se. Okay. Now the 14th question: which of the following is involved in both air conduction and gas exchange? So I stressed on this point a lot. This was a repeated question. Okay, this should take less than a second to answer. Okay, mucociliary escalator. Okay, mucus block is when there is a lot of mucus. Okay, blood vessel dilation actually causes mucus to become warm, even more thin, and it actually moves faster. Okay, increased blood pressure is not related over here. Mucociliary escalator, that is the most common cause of mucus block. Okay, the 14th one: which of the following is involved in both air circulation and gas exchange? Again, respiratory bronchiole. Terminal bronchiole is only for air conduction, bronchi only for air conduction, alveolar sac only for gas exchange. Respiratory bronchioles do both. Now identify structure 1 and 2 slides. Okay. Easy. We looked at this image like a million times. It's easy. Okay. Easy question like this. Really easy. Which of the following cell structure are affected in temporary anosmia? Okay. Temporary anosmia, again, your inability to smell anything temporarily. Okay. So 5, 4, 3, 2, 1. 1 and 2. Okay. The one which is marked 1, that's your type 1 pneumocytes, because you can see how flat the cells are. You can see the nuclei, they're really flat. Okay. And type 2 pneumocytes is the second one. The nuclei are more bulging. As for this, in temporary anosmia, I've told you guys, if the supporting cell is damaged, you have temporary anosmia. Okay. Because these cells regenerate. If the olfactory neuron is damaged, neurons don't regenerate that easily, and you can have permanent anosmia. Okay. Which of the following artery types have fibroblasts in the tunica media layer? You guys did not, may or may not have noticed it, but you will remember it from here 100%. I told you guys how the tunica media has, some have fibroblasts, some don't have fibroblasts, but actually only one of these layers have fibroblasts in the tunica media. Only one. Okay? It's not type 1, it's not type 3, it's not type 4, it's type 2. So that's basically it. And thank you guys for attending it. Do you have any questions the last time? And also make sure to scan the QR code